Thank you all for joining in to celebrate the museum's 10th anniversary. I'm Mavis, your MC for today, from the museum's Outreach and Education team. To commence the symposium, Associate Professor Darren Yeo, head of the Lee Kong Chian Natural History Museum, will be sharing his opening remarks. Associate Professor Yeo began his journey with the museum in 2001, when it was known as the Raffles Museum of Biodiversity Research, serving as a research officer for 4 years. While he later transitioned to focus on undergraduate teaching as an instructor, he maintained strong research ties with the museum. He later returned in a leadership capacity as the deputy head of the museum in 2019 and took over the reins as head of the museum in 2022. He is also an associate professor in the Department of Biological Sciences, Faculty of Science, National University of Singapore, specializing in freshwater biodiversity, ecology and conservation, invasive species, and freshwater decapod crustaceans. Let's welcome the head of the Lee Kong Chian Natural History Museum, Associate Professor Darren Yeo, to the stage.
Morning, everyone. Professor Liu Tan, Professor Peter Ng, advisors of Lee Kong Chian Natural History Museum, friends and colleagues, very good morning to you all. It's my pleasure to welcome you all to the Specimens, Science and Stories, the Lee Kong Chian Natural History Museum's Public Research Symposium. As we continue in this year, to invite you all to continue to explore with us. Now, this is one of several events this year that mark the museum's 10th anniversary. On 6th of May, we held our 10th anniversary celebration event that was graced by President Tharman Shanmugaratnam and Ms. Jane Itogi, in which we launched a new exhibition, Decade of Discovery, that features stories of the museum's explorations and discoveries in our first 10 years. And we also launched a new book, Archipelago of Islands, about the natural and other histories of Singapore's many smaller islands. On 18th May, we held Day of Curiosity, a museum open house that relived a very similar event many years ago that ignited public interest for the first time and calls for Singapore's own natural history museum. That, of course, eventually led to the establishment of the Lee Kong Chian Natural History Museum. On 18th May, we held Day of Curiosity. Over 5 nights in late May and early June, we held Night in Lee Kong Chian Natural History Museum, an interactive event supported by experts from NUS Biological Sciences and Physics departments, where participants explored the museum after hours and learned about forensic science and paleontology. And later this week, our Outreach and Education Unit has a program called Dinosaurs, Extinctions and Fossils that's linked with the school holidays right now. The 10th anniversary celebrations will culminate later this year with one of Singapore's largest dinosaur showcases. Debuting at the Science Centre of Singapore on 11th October, Dinosaurs Extinctions Us is a collaboration between Science Centre Singapore and the Lee Kong Chian Natural History Museum that brings together for the first time 2 world-renowned exhibitions, Dinosaurs of Patagonia and 6 Extinctions. It will feature 33 fossils and 60 models, including a full-scale cast of Patago Titan, one of the largest known land animals. But this exhibition is not just about dinosaurs, it's also about the rise and fall of biodiversity, from prehistoric to contemporary biodiversity, through mass extinction events that have and continue to shape the planet. And this includes the current 6th extinction that we are in now, of which we are both a part of the cause and a part of the solution. There will be information on the exhibition. A QR code will be displayed later, and there's QR codes outside so you can find out more. So it starts on 11th October, but I'm sorry, 11th October tickets all sold out already. So it's popular. Try to get your tickets. Today's event, Specimens, Science and Stories, is the museum's first ever public research symposium. But why do we focus on research? This is something many people have heard me nag and go on and on about. For those who are more familiar with our iconic building, our exhibitions, and our education programs, I think it's fine that people might be more familiar with that and think of us more as a place of natural history storage of specimens and display and education. But the fact is, the museum is first and foremost a center for biodiversity research. It's a natural extension and complement to our scientific collection, which is really the core of the museum. We do local and international surveys and expeditions in all environments, terrestrial, freshwater, marine, across Singapore, with partners in government and non-government, with international partners across ASEAN and beyond. Through this work, we document biodiversity, we make new discoveries, we publish them regularly in international scientific journals, including our own in-house journal, but also recognized scientifically, the Raffles Bulletin of Zoology, which you will see copies of outside. And also a lot of this is reported widely in the media. And beyond the specimen-based research, there's also research in other areas. There's digitization projects, where we are actually doing research to uncover hidden records from Singapore, and we will hear more about that today. And there's also things like the Singapore Red Data Book, where the research is actually translated into conservation documents, things that are a lot more applied and accessible to the general public. Now, with all that in mind, right, the local and international scientific research community already know all of this. So the aim of today's public symposium is really to showcase and communicate to a wider audience the museum's research, both in specimen-based research and non-specimen-based research, through scientific communication and storytelling. Now, in this context, the context of communication and storytelling, research is, of course, still linked to outreach. It's not standalone. So the knowledge gained from research will inform the research, will inform the outreach. And then one example of this is a new stamp release that SingPost is going to have. It's called Critically Endangered Fauna of Singapore, and it's developed in consultation with the museum's researchers and educators. The stamps will be released officially on 19th September, and there will be information, QR code, and things like that, and they'll be displayed either outside or later on the screen. So back to the symposium. Today's program features talks by or with 19 speakers from the museum, including a plenary speech by Professor Peter Ng, our founding head. He will talk about hard truths of a flawed naturalist. At the end of his talk, he's very motivational, right? At the end of his talk, he'll make you want to go out and save animals. If not from extinction, then from him. There are talks on specimens to science, where our museum researchers will share about biodiversity discovery, data collection and documentation, including on insects, crustacea, fishes, mammals, deep-sea expeditions, methods such as DNA barcoding, and very importantly, collaboration and capacity building in this area. There are also going to be a section of talks on specimens to stories, where we tell stories about the specimens as well as the humans and human actions behind the specimens. In this section, they'll be shedding light on ornithology, study of birds in a time of war, about recollecting Singapore, about the history of zoological nomenclature, about sponges in Singapore, and the rise to fame of a very special forest cockroach. There will also be talks about exploring and discovering the great unknown, and finally, the making the most of dead birds. So that'll be quite interesting. And we'll end off with a very special session in Discovery to Dialogue, where museum panelists will discuss and answer your questions, which I think you will be invited to pose today, through the day, about the nuts and bolts of museum research and communication. So I'm really glad to see so many of you here. I hope you all have a chance to see or experience, or at least be inspired by some of the research that we will share with you today. Today's event is jointly organized by staff from the museum's outreach and education team and the research curriculums and collection team, again reflecting the strong link between research and outreach and education. So my congratulations then to Ja and to Jia Xuan and Swee Cheng for the symposium. And my thanks to all museum staff and students involved, speakers, panelists, and event crew, and most importantly to all of you participants for coming today. I wish you all an enriching symposium and an enjoyable time today. Thank you.
Thank you, Professor Yeo. Next, we'd like to invite our advisor, Emeritus Professor Leo Tan, who is one of the two key figures responsible for setting up the Lee Kong Chian Natural History Museum. Over a distinguished 50-year career in science, education, and environmental stewardship, Professor Leo Tan, a pioneering marine biologist in Singapore, has taken on many roles beyond his foundational contributions and current position as advisor to the Lee Kong Chian Natural History Museum. He revitalized the Singapore Science Center as director, championed nature conservation as chairman of the National Parks Board, and inspired and shaped many educators as the founding dean of the National Institute of Education. And for those of you in the audience today who are students or alumni of the Bachelor of Environmental Studies at NUS, you are part of a program that Professor Tan helped bring to life, with the goal of nurturing scientifically grounded environmental leaders and advocates. Even so, these accomplishments mentioned represent only a glimpse into his extraordinary legacy. We would like to now welcome Professor Tan to the stage for his opening remarks.
Thank you. Good morning, everyone. Thank you for coming on a Saturday morning and spending almost a whole day with us. You heard from Professor Darren Yeo, the head of the Lee Kong Chian Natural History Museum, how research and outreach are intertwined. And you will hear lots about the biodiversity research throughout the day through many of our speakers. My opening remarks are addressed at how the public perceives our Natural History Museum. And I think this is important because for too long, ever since I became a young student of biology in the Bukit Timah campus, to the time I became a lecturer in the Cambridge campus, I was always told from the highest authorities to the common man, why are you wasting your time studying biology, studying taxonomy? They'll never get you a job. They're useless. But we know that unless you know what you know, you're not going to make any sense of the animals and plants that you study. And this is what the museum is about. As Professor Darren Yeo said, conservation is a primary goal of doing the biodiversity research. But at the same time, scientists want to know more about the world. But the world wants to know what the heck the scientists are doing that has got relevance to them in their daily lives. And this is where the museum outreach comes in. For example, we found a whale stranded in Singapore in 2015, the year we opened the Lee Kong Chian Natural History Museum. In this today's age of existential climate change, loss of biodiversity, degradation of our forests, etc., pollution, sea level rise, it is even more important to know how the ecology of the environment works. This whale, for example, 10.8 meters, stranded on an island in Singapore. The staff of the museum spent 9 days cutting it up, spent 3 months boiling the bones to make it clean, spent another 9 months to try to mount it, and today it stands in the museum. Is it something for visitors to gawk at or even to say, oh, poor thing, it died from the pollution because of the plastic in its stomach? No, it is more than that. Whales, especially large whales like sperm whales, humpback whales, they store carbon in their bodies. We have roughly about 300,000 sperm whales left in the world. There was once a million of them. And each whale, collectively, these 300,000 whales, absorb into their bodies or remove 200 million tons of carbon each year. Just think about that. Secondly, when they die, they sink to the bottom, deep 200 meters. The carbon stays with them until the seabed animals, the bottom feeding animals, eat their flesh and keep the carbon below intact on the surface floor. They may be our nature-based solution for carbon reduction in the oceans. So studying the whales is one thing, but the application of we know about the biology of the whale helps us to understand how we can protect our environment. And secondly, while they are alive, when they are swimming in the sterile waters of the big oceans, they are pooping. And their poop has got iron and nitrogen. What does it do? It acts as a fertilizer for the phytoplankton and the zooplankton in the oceans. And the zooplankton and the phytoplankton in the oceans absorb 40% of the carbon from the atmosphere and gives out 50% of the oxygen that we breathe in today. So this is one example of how the research complements the outreach program to educate people about the importance of animals. Of course, you'll be hearing lots and lots about tiny creatures today because we do not have many large mammals in Singapore. But nevertheless, even as you walk through the museum, you will not see the research that is going on behind the scenes, but you'll see the results of some of the work. And one example is, there's a picture of a kingfisher in the exhibition hall. I thought it was a waste of money to put up that panel. But I thought about it, and I said, gee, there was once a Japanese engineer who developed a fast-speed train, but each time it went out of the tunnel, it pushed the air, and as the air expanded, it created a sonic boom, disturbing the residents. So the engineer went back to the drawing board on instruction of his boss, and he remembered he was an ornithologist part-time. He loved bird watching. And the kingfisher, he noticed, when he went into the water, the fish didn't even know he was coming until it was too late, because he never broke the surface of the water. And that was because of the aerodynamic shape of its beak. So he designed the Shinkansen, and the rest is history. And that was the birth of biomimicry. So you see the correlation between what an ornithologist was studying about the kingfisher, biology, knowledge about its flight, knowledge about its shape, and the application to everyday life. And this is why this public symposium, the museum hopes that while you are listening to the solid research that it will describe to you, you also think about how will there be applications that will be useful in an ever-changing world where climate change, rising sea levels, loss of biodiversity is an ever-increasing problem for us as humankind. And this is why taxonomy is important, because if you do not know what creatures you're dealing with, how they interact with each other ecologically, then you're not going to be able to solve any problem. And this is why this symposium, I thank all of you, those who have organized it, those of you who have participated, given up your weekend, Saturday, whole day to come. You will benefit and you will profit from thinking about how the presenters are talking about their story and how they are applied in our daily lives. So thank you very much once again. I'm deeply appreciative of being able, of the honor of speaking to you today. Welcome again to the symposium. Thank you.
Thank you, Professor Tan. Next up is Professor Peter Ng, who set up the Lee Kong Chian Natural History Museum together with Professor Tan. Professor Ng served 18 years as director of the museum's forerunner, the Raffles Museum of Biodiversity Research, and 7 years as the founding head of LKCNHM until 2022. Although he has since stepped down as the head, he continues to serve as our advisor and remains highly research active and deeply engaged with his lifelong passion, which is none other than crabs. One of the world's authorities on the diversity and biology of crustacea, particularly crabs in the Indo-West Pacific, Professor Ng has published over 1,200 scientific papers and described close to 1,000 new species in his career of over 40 years. But his contributions go far beyond research. He is a tireless advocate for conservation, biodiversity, and science education. At the heart of his work is a firm belief in the importance of taxonomy as the foundation of all modern biodiversity science, whether you're involved in ecology, conservation, sustainability and climate change solutions, or policy and management. Join Professor Ng now as he shares his fascinating journey of discovery in the world of crabs and conservation. His talk will reflect on species discovery, ethics, and the complex journey of building Singapore's Natural History Museum. Let's welcome Professor Ng to the stage.
Oh, yes, it works. Okay, good morning. I'm surprised there's so many of you on a Saturday morning, right? As some of my big bosses will tell me, all of you should be out there making big money and making big impact for the country and not sitting here listening to a bunch of naturalists and biologists talking about something quite wuliao and doesn't make money at all. So it's very good, actually. Secondly, I need to growl at the head of the museum because I'd rather be spending my Saturday looking at crabs than giving a talk, right? But very hard to say no to people when they are nagging at you all the time to give a talk. So after a lot of nagging by Ja, by Susan, and so on, I thought I might share with you something I was also forced to do 2 years ago. All right, and the title of my talk is actually from a chapter I wrote for a gentleman, which I don't know how to say no to. All right, so about 2 years ago, oh, yes, can I walk? Can I walk around? Oh, I can walk around. Prof Koh wanted to write this book called Peace with Nature. Prof Koh has this vision of writing at least 1 or 2 books every year. So he asked me to contribute a chapter about the museum. I said no. My job at the museum is done. Then about conservation, I said no, because there are better people to do this. And I told him that my main reason for saying no is your title is called Peace with Nature. All right? And everything I do has nothing to do with peace. Okay? How can you ask me to write something in which my whole career has been drenched in blood? Because that's what most of my friends and enemies say. All right? You kill animals for a living. All right? You are no saint. So, of course, I kept saying no to him, to Lin Heng, to Sean. And finally, he called me and says, no, you're writing a chapter. All right? And for those of you who have worked with Prof. Tommy Koh, very hard to say no to him. All right? In fact, the museum that you have seen and we've been talking about is also his fault partly. All right? Because 20 years ago or 25 years ago, when he visited the old version of the museum, the ZRC, he suddenly inspired, said, finally I can have my natural history museum. I've got art museum, I've got Chinese culture, I got toy museum. I always wanted a natural history museum. Okay? Peter, go and build one. And my first reaction is, who's going to pay for it? Of course, you can't say no to Prof. Koh. And 10 years later, with Prof. Leo Tan's help, we actually built the museum through a series of interesting events. But that's another story. Okay? So he forced me to write a chapter. So I thought, well, since you asked me to write a chapter, and I have no choice, I might as well tell the truth. Right? Prof. Leo Tan is an optimist. He believes the future, there is a good future. I'm a pessimist. But since I don't like to lose, I'll be an optimistic pessimist. So I've been asked to share a few things, basically from the theme of the chapter I wrote for Prof. Tomiko. And first thing I have to say is whatever I'm going to share with you is my personal perspective of how the world is. All right? I've been told that I am biased. I see things through a different lens. All right? I don't see things through the official lens. All right? Prof. Tomiko calls me an iconoclast. Now I quickly look it up what it means. All right? Basically, it means a troublemaker. All right? So I said, all right, fine, I can live with that. So I'll tell you 5 things very quickly, since I only have 30 minutes. All right? So the first accusation, and which I accept is true, is I've got a lot of blood in my hands. I'm a taxonomist. I'm a naturalist. I hunt animals for a living. I try to find out what they are. In order to find out what they are, I need to look for them, catch them, kill some, put them under the microscope, and do other bad things with them. But basically, is they have to die. Okay? If you go to the museum, you see tons, thousands, tens of thousands of dead bodies collected by my predecessors of a similar kind over the last 150 years. All right? And I've been frequently lambasted by different people. I'm a cruel bastard. All right? How can you live with yourself? I have never claimed to be a saint. I don't think I qualify. And I certainly cannot be a vegetarian. My gut doesn't allow me to. All right? And I'm none of the above. Am I in the dark side? If I'm in the dark side of biodiversity, then so be it. All right? Because in the world we are living in today, and I don't have to tell you, all right, it's far from black and white. I like looking for new, rare, strange animals, okay? I like to go to places that people ain't seen before. People have not hunted before. Because that's where all the cool and strange animals are. And in order to do that, once you catch all these unusual organisms in a myriad of ways, you have to preserve them so that they can be studied. And not only that, we are dealing in the science of taxonomy and biodiversity. It means that whatever samples you have to stand the test of time. Other scientists can come along, check these bodies and say, no, Peter, you're wrong. It's not this species, but it's that. No, it's not A, but it's B. No, it doesn't do this, but it does that. And in order for science to be objective, you need dead bodies there so that people can check. I've been told, photograph cannot. My son's Photoshop skills are so good, I can't tell whether he creates 10 new species based on one specimen. All right? And most of you have seen the power of AI. All right? A photograph cannot. Trust me, I saw it. I know I saw it. I don't trust anybody. I trust a dead body because the dead body is objective. Now, it is interesting, it's fun, it's scientifically rigorous, but it presents every taxonomist, every hunter with a terrible moral dilemma. All right? You have to kill the animal. Of course, you can kill it the most humane way possible. You don't kill them by the thousands, all right? But you have to kill some, right? Is it cruel? Of course it is, right? But what's the alternative? Nobody can come up with one, all right? And very often I get these lectures from people which love to eat steak. My best is from a student. Oh, Prof, you shouldn't do that, all this. I say, and do you eat? Yeah, I like chicken. But you kill chicken. No, Prof, I don't kill chickens. I buy mine from the supermarket. And how does that allow them to justify that they don't kill? This audience here, I can tell you a certain proportion of you are pure vegetarians. Most of you are semi-vegetarians. Most of you are omnivores. All right? So first thing is, I will admit, I'm a hypocrite. To do my science, to implement the things I need to do, I am a hypocrite. Prof. Tan says, to do conservation, we need to know the animals. Of course we need to know the animals. The Chinese have an old saying, very ancient saying. The first step to wisdom is to call things by the right name. So we need to know what they are. Mantra, you cannot conserve what you do not know. You make a case to conserve X or Y, we need to find out what the heck they are first. All right? As best you can. This is what I share with my students all the time. A very famous saying in a cemetery in World War I in Verdun. All right? The truth of all conflict. You can't fight for your freedom and say everybody lives. Some must die so that others may live. No difference in all of us here interested to conserve biodiversity, keep nature alive, keep the green world we have. Something has to give. How much? That's for you to decide. All right? You don't have a choice. Okay? You can't have a free lunch. There's no such thing as a war with no casualties. So, over the years, I've become quite blunt. Okay? I don't ask for forgiveness. Just understand why am I doing it. Okay? Many of you will do the same thing and pretend you are a good person. No. At the end of the day, we have to kill things. You cannot be a totally good person. Even Marcus. Right? He's one of my curators who tries not to kill things. Right? But sometimes, right? So, there are no saints. Second question I get very often from my peers and friends and colleagues is, why the heck do you play with crabs? All right? What drove you to crabs? Why do you have this inordinate obsession with crabs? And it's a very interesting question. And every time I ask myself I'll tell the wife why did I marry you? And she gives me that very grumpy look, all right? It's a love affair you pick up somehow, somewhere along the way, and sometimes most of it cannot be explained, right? And it cannot be explained. I seem to like crabs, and it's been a journey for the last 40 years, and it's been a nice journey, all right? It is what it is, okay? Some of you like other groups of animals. Some of you like butterflies. A lot of people like butterflies, all right? YY likes birds. I don't like birds. Well, I like birds on a dish, all right? She likes to study birds, okay? Each of us develop our own passions with different kinds of plants and animals along the way, right? Some because of parents, friends. Some, it just happens, all right? My parents told me not to waste my time studying animals. They wanted me to be a doctor. I refused. They wanted me to be a vet. I didn't become a vet. They wanted me to be a lawyer. I almost became a lawyer, all right? But this is more fun. And this is a great journey for the last 40 years. I have gone around the world chasing after all sorts of animals mainly. A few times I chased plants for the fun of it. Right? So Southeast Asia, of course, you see all the red dots. The red dots are places that I've gone to and done bad things. Blue dots are where I've sent my mercenaries to do my bidding for me. All right? Or they have gone to museums to look at specimens. So we're looking at dead bodies, but we didn't kill them, so it's not our fault. All right? And it has been a wonderful journey because you get a chance to see strange places and see the world. All right? First thing I need to be clear is, a lot of people think that I'm a marine biologist. I am not a marine biologist. All right? Even though I do a lot of things which are marine related. Okay? I'm not a freshwater biologist, even though I created some freshwater biologists like Darren. All right? He started his career working on freshwater shrimps. Then he decided that shrimps are not nice, are not so easy to work with. Then he went to crabs. Crabs have interesting reproductive structures, so that gets him excited. So he has been doing that for a long time. Then he carried on working with freshwater crabs and freshwater biology. But I'm not a freshwater biologist, all right? My life's career has been very simple. I follow my crabs. There are crabs on high mountains, I go there. There are crabs on lowlands, I go there. There are crabs at 2,000 meters, I try to go there. All right? Wherever the crabs are, my expeditions follow. All right? And other people go along and catch whatever else they want. All right? And I hunt for the animals. When I was younger, I could still climb waterfalls. Now I can't climb waterfalls. My knees won't allow me to. All right? Actually, I still think I can, but the wife says I cannot climb because I may fall off. And I've been working with people, going to and sending people all over the place to all sorts of habitats, from caves to swamps, to peat swamps, to ocean depths, to coral reefs. And it's been a wonderful journey. You see all sorts of wonderfully strange crabs and other animals. All right? And this eureka moment when you find a crab or even a fish. In the old days, when I was younger and more energetic, I played with fish. All right? Then I did get bored. The fish are still quite nice. All right? But then the people I trained up, like Heok Hui and Kelvin, turned out to be better than me at fish, so I left them to it. All right? So I went back to my crabs. And the eureka moment, the feel of finding something new or rare, all right? Something that has not been seen for 100, 200 years. I tell you, it's a fantastic feeling, right? And once you know, it's great fun. And yes, that's Heok Hui and Swee Hee, all right? They were students with me a lifetime ago. Despite all my best efforts, I couldn't get rid of them. All right? Yes. They were in a peat swamp collecting fish. And the water is that color. The excitement of exploration and looking for rare species, crabs inclusive, is that very often we don't know what we're going to find. And we begin to realize after a few years that actually, if you think you know what's out there, most of us, I can tell you, who has been in the field, will tell you, you are terribly wrong. You will always be surprised because there's usually much more out there than even you can imagine, right? And the surprise is what keeps the blood flowing. It's very exciting. Today, I can't catch so well. Fortunately, I've got lots of hunters out there who show me and catch all sorts of interesting things for me. And when they show it to me, they know how to get me excited. Oh, show me a crab I've never seen before. Show me a crab, I don't know what it is. Right? The excitement drives me. I love crabs. Okay? Chances are I'll drop dead in front of a microscope with a crab underneath it. Right? But that's a good way to go. Right? But I will never finish my job because there are too many of them. Right? So each generation will find its comfort zone. Each new generation of crab people will carry on. All right? Another question I get is very often from government officers. Oh, you discover so many animals, so many crabs. What's your favorite one? What's your most important one? What's your most memorable one? Right, Yun Ping? And my answer is always the same. My best discovery is the next one. Okay? I find something cool. I work on it. I report on it. After that, I move on. I go do something else. Right? And it's the nature of science. You cannot be stuck on one thing for too long because the next thing is more exciting. And you move from one to the other. All right? I mean, very often they keep telling me that these two silly crabs should be my most important discoveries. All right? This way song, home is where the heart is, right? Well, 1985. How many years ago is that? 40 over years, okay? 40 over years ago, when I was young and climbing around Bukit Timah Hill, all right, I found crabs running around in a storm. I picked it up, took it back to home, had a permit at the time. All right? Who was the... Governor of Parks then. Yeah. They gave me a permit. I went up there, caught them. In those days, getting permits so much easier. Just fill up a simple piece of paper, have one phone call, oh, Peter, go. All right? The rangers, they'll see you, I know you're coming, they go. These days, a lot more paperwork, I'm told. I went up there, caught the crab, sat on it, worked on it. Oh, it's a new species. Wrote it up. All right? It was just along the way sort of discovery. All right? Another new species. Another interesting animal found. A few years later, all right, this pesky art student who's not here today. Mr. Lim is not here, right? Why are you laughing? Why? Okay. This geography student, art student, says, oh, I saw this strange crab in Nee Soon Swamp. All right? Is it the common one? I look at it, yeah, probably a color morph, a mutant. Like you happen to see one odd one. All right? And that was the crab at the bottom. All right? But then since it was found, he showed me a beautiful photograph, it was a nagging feeling. What if it's not an aberrant one? So after a bit of pondering, I said, Kelvin, let's go to Nee Soon. All right, we spent several New Years down there. Right? You need to go there, catch them in the middle of the night during when there's no moon. Very annoying crab. Finally, we caught them. And of course, Kelvin was right. It is not the common one. It's actually a different species. And annoyingly, it's also endemic to Singapore. So you have one endemic, Johora singaporensis, one endemic, reticulata. Why am I so sure they're endemic to Singapore? Because I've been killing crabs all over Johor, Batam, Bintang. All right? Now I sound like Phua Chu Kang. And beyond. All right? We have hunted crabs all over the place. And these guys are not there. We have done the genetic studies required. We know they're endemic to Singapore, which is very good. When we discovered them, we thought, well, fine, more species, a few species endemic to Singapore, very nice, all right? And, of course, in the years ahead, as the world changes, all right, I've been told by some authorities that the climate is changing, all right? Even though some powerful people think that climate change is a hoax, right? The world is changing. And suddenly, conservation of freshwater animals becomes higher priority. And one of those meetings back, how long ago was this, Darren? When they decided to make this fellow famous? 20 years ago? Yeah, so when IUCN were drawing up the list, they said, all right, instead, we cannot just worry about birds and butterflies. All right, let's throw a few invertebrates in. Let's throw in a cool invertebrate. Let's choose a freshwater crab. And for whatever reason, this crab went in. At the time, this crab was also disappearing from some of the places where I used to find it. And suddenly, it hogged the news. All right, we have one of the world's most endangered animals in Singapore, and it's about to die. Then agencies and parks all jumped in, all right? Wanted to conserve it, had to make sure it doesn't become extinct, all right? As far as I'm concerned, when I talk to some of the authorities, they say, oh, Peter, why do you want to conserve this crab? Why are you so worried it becomes extinct? I say, I'm not worried. If it becomes extinct, I just blame you. All right? I mean, I discovered the crab. If it becomes extinct along the way, it's not my pasar, all right? Government agencies protecting the parks. Some of the crabs were found in MINDEF even. I told the colonels, well, blame MINDEF, law, blame NParks, law. All right? Right, Jim? Right? Of course, they were not very amused, right? And along the way, NParks now has a very robust, very nice program to keep these crabs alive, right? To make sure they don't die out, all right? And to spend lots of taxpayers' money to ensure that an endemic animal survives through the crisis we are in. Right? Jim won't tell me how much, but it's a lot of money, right? Now, and this is part of it's not my book because Jim's boss one day caught hold of me and says, Peter, why are you taking such a nonchalant view about the crab? We are trying to save your crab. You should be more enthusiastic and grateful. I looked at him and I said, no, it's your problem, not mine. And the reason is very simple. I'm the discoverer. My job is to find strange animals, all right? I'm good at taxonomy. I know my crabs very well. I've been around the world. I see new species. I can tell you they're different. I can tell you why they're worth conserving. But the choice to conserve has to fall to a different body, not me. Am I being selfish? Partially. But there are people which are very interested and very capable of doing it. They should do it, okay? I'd rather spend the rest of my time looking for other crabs, looking for other strange things around the region, right? Am I irresponsible? Yes. All right? But I'm not Superman, okay? I have 24 hours a day, and if there are better people out there to save the crab, they should be doing it. The government agencies want to dedicate resources and train people to do it, carry on, all right? I think Darren and his team are involved in some of these conservation programs now. Very good, all right? But to me, it's just a crab. One of many hundreds I found. Just coincidentally, 2 of them are in Singapore. Right? Conservation of any endemic species is a country's responsibility. The people must want to conserve it. Okay? The agencies represent the people. The ministers, the elected officials we have. If they don't want to conserve it because it's too expensive, the crab dies. Is it my fault? No. As a Singaporean, I'll feel sad. As a scientist, I'll just say, c'est la vie. Now I sound irresponsible. Look, there's so many habitats, so many crabs, so many animals out there, all right? Too much to do. Too little time. Finding new species and strange species is great fun because the diversity out there is surprising. After studying crabs for 40 years, I can tell you, we are beginning to chip away at the tip of the iceberg. What 40 years of study tells me one thing, I'm ignorant. What I think I know, what we thought we knew, is incorrect. We know very little. So there's so much to do. And the worst part of it is I'm running out of time. All right? I'm in the wrong half of my life. Okay? Unfortunate, but that's life. Yes, the one thing I refuse to do for Prof. Tomiko, write about the museum. Okay? As Darren said, okay, I'm the founding head of this place. Not this place, sorry, this is not my place. The other place, right? Am I proud of the museum, proud of everything is done? Actually, the answer is this. what can I say? All right? Building the museum together with my team of folks and Leo and all this, finding the money, all right? Prof. Tomiko is a very annoying man, all right? You can't say no to him, all right? He tasks you to do all sorts of things, and I always ask him, Prof. Ko, where's the money? Oh, you will find it, don't worry, the vision is correct. That means we have to find it, lah. All right? And that sense of optimism and, shooting arrows, he's very good at it, all right? So, but yes, the museum is important, okay? This phrase from John McCain, all right, is very nice, all right? Of course, some people think that this guy is a useless person who got caught by the enemy, all right? I don't think so, all right? I read his book. It's a very inspiring book, all right? My biggest sadness about him is that he had to compete with Obama to be the president of the United States that year. And that was a lost fight, all right? Otherwise, he's a good man. His years in the war, his years as a POW, and the years in the Senate, in the Congress, actually was good, okay? The museum is bigger than Leo and me. It is a national institution and an institution for the whole of science. It is a challenge worth taking up. Tomiko was correct. Leo was correct, all right? It is a worthwhile challenge. It is an important facility. But not something I'm totally proud of. It's an important job to do, but it's not the same, right? First thing is, I don't really believe in legacies, okay? All organizations, all institutions that are built by people, by groups of people, they are built for a particular reason at a particular time, all right? The reasons for its existence and the roles it will play will change over time. How it will change will depend on subsequent generations of leaders, all right? As far as I'm concerned, all the problems of the past or the challenges of the past were resolved partially when the museum was built, created, and what it will do will depend a lot on the new generation of leaders, the university, and the people who support the museum. And I can only hope for the best. Nothing is forever, all right? I've been through 4 vice-chancellors, 4 or 5, I lost track already, all right? God knows how many deans and heads, all right? I've seen some very good heads and deans ostracized by later heads, all right? I think they were good. The successors said they were useless, all right? Every generation judges the past in different ways, all right? So don't believe in legacies, okay? We just hope for the best, that the people leading it will do the right thing, okay? My only hope, new museum staff, okay? You will do the right thing, whatever right it means. It was fun building a boring museum, all right? Everybody tells me this is a piece of rock, all right? The building next to us, the big museum conservatory, wow, so it's beautiful. I said, sure, all right? Cultural centers, grand, sure. Yours is just a rock, thank you. This is a very practical rock. It saves a lot of money building this, all right? At the end of the day, the building, the facilities, is just a shell, all right? What value add is to it, like it or not, are the people. How famous, how great the museum becomes, how many millions of dollars it brings in, how much fame it goes around the world and the region, all right? How many papers it get published is actually irrelevant. At the end of the day, is these living bodies that are in there, what they do, that will keep the sense of the museum alive. It's the people. The people who build it, the people who populate it, the people who use it, okay? I disagree with some of my bosses, okay, where they think that humans are a resource. They are a resource, they are also an asset, all right? To me, the people are more important than the KPIs. But that is a matter still under discussion. Will I do it again to build a new museum if I had a chance again? Probably not. Very tiring, all right? Leo will probably say yes. Yes? Yeah, he's a good Mandarin and a good champion, all right? It was a hell of a journey, all right? It's something you do once, you don't want to do it again, all right? Even for good reasons, all right? For those who have been with me a long time, and all of which are old, but Kevin is not here, so I cannot insult him. All right? The old museum had a sense of family, which was very nice. It was a small group. The new museum is a much larger entity, and therefore we hope we can do a lot more good, which is true also. All right. Okay, running out of time. Last thing, the museum is now safe. Things have been built, everything is nice and comfortable, or not comfortable, okay, all right? The ship, as they say, is in safe harbors, all right? The Natural History Museum, the challenges of the old Raffles Museum and ZRC are over. The ship is in safe harbor. That's the expression I heard from one of my colleagues. This is a nice expression, okay? And my answer to the colleague who told me this is, I say, yes, you may think the ship is in safe harbor, and it seems to be the ship is in safe harbor. But the problem is, I think ships are not supposed to be in the harbor. The role of big ships is to ply the oceans and the seas, take the risk of typhoons, storms, tsunamis, all right, pirates, all right? Because their job is to sail around doing things risk inclusive, all right? Be in a safe harbor is not where the ship should be. It should be where the ship is for repairs, and after that you go out again. I make a few comments for a future which I cannot even imagine, all right? I tell a lot of my colleagues to guard themselves against this thing called hubris, all right? Because hubris is, in Greek mythology, is a sin when you think you're smarter than the gods. You think you know more than the gods. You think you know more than the big chiefs. That's a very dangerous sin, all right? And hubris is what brings empires down, bring organizations down. It's a very dangerous thing. I tell myself all the time, the older you get, the more likely you get hubris. And the penalty for hubris is another god that threatens retribution. I always like this crap from Singapore, all right? In 1980s, I called this Josisama nemesis, all right? Because the stupid crap had the audacity to bite me. For those of you who work with me, you know I'm very dexterous with crabs. I can handle even the big crabs, small crabs. I can handle them, and they always ask me, boss, how come they don't bite you? I say, because the kaki lang, you know. But occasionally, one stupid crab bites you, all right? So I name it nemesis, all right? Because it's a presumption you can never have. And nemesis usually involves a lot of terrible things happening. So for those of you in the museum and working with the museum, please guard against hubris. Please. The museum right now, as it is, doesn't have challenges of the old. The danger is, it's a danger, we don't have it yet, the danger is that we become complacent and comfortable, right? The challenge is that we must never become too comfortable, right? And one of the things we always joke in the museum still, the museum is a place we do impossible things, because they thought that the task of building the museum that Leo and I had was impossible. Actually, it was not impossible. All right? It was unimaginable at the time. Not impossible, right? So to do more impossible things would be something the museum should do more of, all right? Or as another manager at NUS told me, you all do a lot of stupid things. I say, yes, thank you. Stupid is a matter of definition also, all right? We chase money. We chase money to build a museum. Yes, Susan, I know I'm not supposed to put the dollar note down here, all right? This is illegal. I'm supposed to put a word here, not legal or whatever, all right? But I didn't have time to do that. Okay, you're not supposed to put money on any slide, all right? We chase money for a lot of reasons. We chase money for research, to build a new building. As we chase money, and we chase money still, always ask yourself, what's the money for, right? Money can corrupt the soul totally, and corrupt your mission totally as well, right? There's a lot of push in the universities now for what they call fab science, all right? High impact science. Make the university famous, make Singapore famous. The problem with the science that we do, biodiversity and natural history, is we work on the slow burn. That silly crap I found in 1985 became a commodity of high conservation significance in the year 2010 or 2005, all right? Slow burn. 30 years before it became value, all right? And I'm told it's a one-off. It's not a one-off. Biodiversity sciences, the results you get most of the useful things that will come are on slow burn. Okay? Today, we tend to measure science on this very short window of 5 years based on impact and so on. The best one I ever heard, one of my, okay, cannot mention name, asked me, why do you keep citing Linnaeus? Linnaeus was 1758. What kind of science are you doing? You're still citing 1758. You know, who started citing 1758? I said, we do. What kind of science are you doing? Our science, because Linnaeus is the father of taxonomy. A lot of the animals discovered were by Linnaeus. Of course I cite Linnaeus. Why? What for? Because he discovered it. He doesn't understand the cycle of science that sometimes it doesn't work on 3 years, 2 years, 3 years, or 4 years. All right? We work on long times. And that's frustrating. So don't get too caught up in this, what I call fab science, okay? It's this very famous expression, okay? A diamond is valuable, but in a desert, it's totally useless to you. Okay? We tend to count sometimes the wrong things, all right? I call this the corruption of science. I'm told that's an impolite way of saying it. It's okay, I'm an iconoclast, I can say whatever I want. And in this strange new world, Leo keeps telling me we're living in a transactional world. Yes, we are. It's a transactional and a mechanistic world, and more so as we rely on machines and numbers to tell us what we do today and do next. Okay? Nothing wrong with that. They are important. All right? But you always have to remember that at the end of the day, you have to do what you believe is right for yourself and the long term. And it must be fun. All right? Many of you are here because you're not really academics or scientists doing pure biology. You're here because it's nice and it's fun. All right? I should tell you to go out there and make money instead. But you rather spend your time here listening about animals, natural history, and science. And that's the problem with human beings. The problem with human beings, I can't make them into computers and robots. They're very annoying. Not yet. All right? And since remember Hebrews 11:1. I won't tell you what it is. Right? We need to believe. My passing slides, okay? I've been doing this for 40 years. Chasing down animals is great fun, okay? This old friend of mine from Malaysia, Prof. Yong Hoi Sen, in Kuala Lumpur, PJ, told me a long time ago when I was a young man, you see how young I was then, not so fat. All right? I was asking him a very simple question, a young scholar, Prof, so when do you think I have enough to write a paper, to publish? He looked at me, when you got a good story to tell, you write. It's big, small, doesn't matter. As long as you enjoy, you think you got a story, you write. All right? It gets into the literature, your peers will decide whether it's good or bad, whether they think that you are a bum or you are a scholar. All right? Publish and be damned. All right? If you are doing good science, you'll be fine. If you're writing rubbish, everybody will tell you that. Right? You judge, you move. All right? Science-driven, it is driven by passion. All right? Don't worry about rewards. Okay? Share the knowledge. That's your job. Whatever you find, you discover, let the world know. Let the world decide whether it's useful, interesting, or otherwise they want to take anything with it. Not for you to judge. Okay? It's advice I've been using for the last 40 years. All right? Each time I got a story to write, nice story to tell, I'll write it. Should you do that? I hope you do. Okay? Okay, Darren, please read this. Danwei also. Okay? Remember, the ship has to sail. Right? Or Star Trek says to go where people have not gone before. All right? So do a lot more silly things. Take more risks. All right? One of my favorite characters from literature is Don Quixote. All right? He's a madman who wanted an impossible dream. All right? But that passion drives a lot of things. All right? And it's not just for the management of Darren and Danwei. It's for all the young curators there and there. All right? It's to push the boundaries. Right? And you should do it with God's speed. Can you do it? I don't know. I hope you can. All right? Thank you.
Thank you, Professor Ng. Now let's proceed to the first session. In our morning session, From Specimens to Science, we will be focusing on the bread and butter biodiversity work that we do. Our museum is just a 5-minute walk from here, and while the first floor and mezzanine are publicly available, today's session will explore what's hidden away on the other floors. Our speakers curate and manage the extensive collections stored in the museum's upper floors, so you will get to hear about what goes on behind the scenes during species discovery, museum work, and conservation efforts. Each speaker will present for about 12 minutes, followed by a short Q&A session right after their talk, in which you can raise your hand and we will come to you with a mic. Do also note that there will be a panel discussion after all the talks at 4 p.m., so if you have any questions you would like to ask the museum, you are more than welcome to submit them via the QR code up here. We will show the QR codes a few more times throughout the symposium. During the talks, please note that flash photography and videography are not allowed. For our first speaker, Dr. Hwang Wei-song, he is a senior curator of insects at the museum. He specializes in true bugs, particularly the predominantly predatory assassin bugs. His research work includes narrowing down on the total number of insect biodiversity present, especially in Singapore and Southeast Asia. This talk provides a progress report on how far we have come in getting a sense of this number and the immense task that lies ahead. Let's welcome Dr. Hwang to the stage.
Hi, very good morning, everyone. Thanks for coming over. It's a very great honor for me to be here to share with you some of the results that we have been going through for the past 10 years. Right? And I didn't know that what I wrote for the synopsis is meant to be read out here. But knowing that, it's good to know that part of what we do here is actually trying to share, right, the research that we are doing here. I would like to invite everyone to think about how biodiversity looks like to you, right? How would you imagine biodiversity when we talk about that term? And one of the examples here I'd like to see is that this is a very typical kind of picture that you see when you do your Google search, right? And I'm biased. I'm an entomologist. I work on invertebrates. And to me, this is not a very good picture or representation of biodiversity because it's pretty much only 3 major lineages of organisms that you see here, right? You have the mammals, you have the birds, you have the plants. If you squint, I think there is like one octopus somewhere and there's a dragonfly another way, somewhere else, right? So to us, working on invertebrates, this is not how we would see biodiversity, right? Then the question then becomes, what is the best way to visualize biodiversity in a sense, right? When we talk about the term. And I'd like to invite you guys to think about it. And one example that we have seen is this thing called the speciescape that was drawn out in early 1990s, right? To talk about what we have, right, in terms of total number of species. And this is basically trying to scale up according to the number of species that we know that has been described out on Earth, and trying to see how many species are there altogether, right, depending on the groups. And this is based on the different major phyla, right? So you have insects, you have plants, you have turtles, and you'll notice that, of course, right, those that we are most familiar with, the birds, the mammals, they're not that big, right? And what is big is actually more of the invertebrate groups, right? So your mollusks, the mites, and the insects. Another way to think about biodiversity is, of course, to look at the biomass, right? The total number of organic matter that exists, that's locked up in living organisms, right? So here, things look a little bit different, right? Livestock, actually, basically animals that we grow that we can eat, actually is quite significant in terms of biomass, right? There are more chickens out there in the world than there are humans, and there are more chickens out there than ever in the whole of evolutionary history of the Earth, right? So we grow a lot for produce, right? But here, once again, I'd like to point out the fact that we have arthropods, right, which is all your animals with exoskeleton. that actually takes up quite a bit of space, right? Not just in terms of species numbers, but here also in terms of the amount of organic matter that exists in that sense. Now, one of the things that you don't often see is the fact that biodiversity actually is messy. When we get biodiversity samples, when we go out there to collect, it's a mess, right? And the animals don't tell you what they are, right? You have to sort it through. Those people that have actually gone through insect sorting, for example, and one of the things that we do here in the labs is actually go through Malay stripe samples. There's a lot of colambula, there's a lot of springtails, there's a lot of mites, things that most of the time we don't even pay attention to at all, right? So things are messy. So let's bring the perspective back to Singapore per se. For my talk today, I would like to just concentrate on Singapore, right? Because we have been actually spending quite a bit of time working on the biofauna that we have. And for me, of course, it's insects, right? So very broad introduction of what we have here, right? The area, we're not a very big place, right? We have grown actually quite a bit since the founding of modern Singapore in 1819 because of land reclamation and so on and so forth. We drained some of the swamps. Interestingly, 15% of our land area is taken up by Changi Airport. There's not many countries out there that is 15% airport, right? So we're not that big. More than 90% of the original forest cover we had was already lost by the first 100 years, right? So you can very well imagine that the fact that the landscape change actually happened very, very quickly, in a very short span of time. And what we have here as of now is actually very different 200 years down the road, right? And not only that, the primary rainforest that we have left on land is less than 1%. It's, you can squeeze it very easily into Changi Airport, right? So what we have left in terms of natural environment is not a lot. We have a lot of people, we have a lot of economic needs as well. So all of this is, of course, constraint for space, right? So the natural areas that we are most interested in as a naturalist for the terrestrial side is really pretty much the green areas, right? So you have your central catchment area, you have Bukit Timah, you have your western catchment area quite a bit as well, but that's less accessible, right? So not much space, right? But we still have stuff, right? We still have biodiversity. In terms of biodiversity, what do we have, right? So as a beginning introductory slide, I'd like to show you some numbers. For native plants, we have about 2,654 species, right? So the botanists have done a great job running around in Singapore. I don't know whether you guys have met any botanists. I have very high respect for them. They're very methodical. They are very gung-ho. They're better than zoologists in organizing the stuff, so I trust the numbers. Interestingly, because Prof. Peter Ng was talking about the fact that we kill, right? Botanists will laugh at us because they don't need to kill. Right, they can actually just sample the flowers, sample the branches. They have something called the viva type, which means that the original specimen that they have described, the species, could still be standing there. And we do have actually some viva types in botanic gardens. Right, so plants, we're actually doing a pretty good job. Birds, we have about 460 species. Once in a while, we get some migrants. So those are actually pretty good numbers as well. Reptiles and amphibians, 138 species. Freshwater species, 102 species, and you'll hear from Dr. Tan Heok Hui later on that actually we still have some nice surprises. And then mammals, of course, 91 species, and that actually includes those that we have already lost, right? The extant ones is actually much smaller, right? So these are good numbers that we have when we assess biodiversity here. And then, of course, as an entomologist, my question is, what about insects, right? What do we know about them? Well, some insects we actually still do have quite good numbers. For dragonflies, we have 118 species. Butterflies, of course, everybody loves butterflies. We have a lot of people looking out for them. Every time there's a new one that pops up, it comes up in Facebook the next day, right? And then, of course, we have our very hardworking ant curator, Dr. Wendy Wang, who published recently about the fact that we have actually more than 400 species of ants, right? So for some of these groups, we do have a nice figure. But that's not all it is, of course, right? There's a lot more other different insects altogether. And to get a handle of this, as simple as the question that we have, how many species of insects do we have here in Singapore, it's actually a more challenging question than it looks, right? And that's one of the I would say the preoccupation of mine, it's not an obsession yet, that I have in the past 10 years to try to come out with a nice estimate. Right, so to do that, of course, we have to go out into the field and set up all sorts of traps. The one annoying thing about insects is that they're found all over the place, right, when you go into the forest. Right, they're up in the canopy, they're in the leaf litter, they basically make you work for it. Right, so when you go out into the field as an entomologist, you can't get much sleep because there are those that are flying in the day, there are those that are flying at night, and you want to catch them all, of course. Right, so we deploy all sorts of different kind of methods, try to come up with as many ways to catch different kind of insects as possible. And then because of the sheer immensity of the numbers that we're dealing with, right, we are not able to do the traditional morphological identification for all of them. We want to have a shortcut. We want to have an estimate, right? And here comes, of course, DNA technology that has been well established in the past 10 to 15 years. We use molecular barcodes to try to have a rough estimate, again, right, of what we have. And we've been doing this, right? So somewhere in the northern part of Singapore, where there are still elephants or tigers roaming around, we actually got the chance to do it. We did a very extensive sampling over there from 2019 to 2021. But just with 1 month sample, we demonstrated that this method works, right, in the sense that we can actually get a rough handle of the number of species of insects here. Right, so with 16,000 over specimens processed, right, we use the genetic threshold, right, to give an estimate, and that is just within the small little area that we call Mandai, right, we have about 2,851 putative species based on the molecular estimates, right, and that was a proof of concept to say, hey, we can actually have a means to have some idea of the insect diversity here. Now, all that is well and good for us. Along the way, we actually had some collaborators as well, right, that were able to basically repeat the same kind of sampling across the world. One of the interesting findings we had, right, when we combined all our results, was the fact that the top 20 insect families across the world, regardless of your habitat type, regardless of continent, it's actually more or less the same, right? That was actually quite an unexpected result. The second part of this finding that was interesting was that most of us have no idea what they are, right? And this is actually very sad, right? So one of these species that we have here, the Cecidomyians, these are the gall midges. This is by far, anywhere you go, there will be gall midges that are there, that are found, right? It's sad because basically what it's trying to say is that nobody wants to work on them, right? Anywhere you go, regardless, nobody has the interest because they are not that easy, right? They're actually quite hard to identify. They're quite boring. They're not very descriptive. They're not so showy, but they are in great numbers, right? So that's a dilemma that we have to deal with. So we got a second chance, right, to refine this and to have an even better idea of what we have. We actually got the chance to join in to the CCNR, the Central Catchment Survey that's ongoing right now, with some collaborators on the DBS side, the Department of Biological Sciences. We actually sample across different kinds of forest types, all the way from primary rainforest to secondary forest to more disturbed woodlands. And this time around, we did a slightly different kind of molecular approach, but again, the whole point of this is actually to try to figure out how many species of insects we have. Right, so with the collaborators, we sampled for 1 full year, right, across all the different landscape gradients. I wouldn't go into detail for this, but we did a slightly different molecular approach just because of the sheer immensity of the numbers of the process. We weren't able to do it one individual at a time. We basically crunched them up into a DNA soup, right, so that we can have some estimates. And the nice result, and this is not published, this is fresh. This is not even fresh off the shelf. This is still in the oven baking. We still have to put it back to refine it still. But I'm happy to share that right now we have 1 year's worth of sample. We have incredible diversity within our forests, more than 225,000 unique DNA sequences. When we look at molecular clusters, which is our species estimates, we have 140,000, which are the putative species based on the genetic information, right? So just to put things in perspective, the only other country that has published the total number of insect species they have within the country is Germany, And they've been doing this for good over 20 years, and they have 34,000 species. Right, so this was actually a quite nice finding, and we're still refining this. Right, so something skipped. I think I have some slides that are missing, actually, but it's okay. What does all this mean to us is the fact that, why does all this matter, right? Whether or not we have 140,000, whether we have 100,000, why does it matter? The reason is because every species counts, right? The fact that we want to know every one of them is a long shot. 140, way beyond our my lifetime, definitely, for sure. But I'd like to share a few examples about why species matter, right? Two of them. One of it, we actually discovered a new species in NUS. Right, this was only done, published earlier this year, but the work was actually done 2 years ago in 2023. I invited the plant bug expert for Southeast Asia, came down, took a look, found one tree, found the merit. We describe it. So now NUS is the type locality for this species of plant bug, right? And the nice thing about this is that it is actually the host plant, at least. This plant bug is found on one host plant. The host plant is all over the place in Singapore, actually. It's a sea hibiscus, right? So it's actually found mostly in coastal regions. But the fact that we have them actually a lot of places goes to show that there's still a lot of things that's to be discovered that are actually right under our noses. Okay, so, I think this got misplaced, that's why. Never mind. Let me move along with the slides, right? So when you look at terrestrial biodiversity in Singapore, if you add all the insect species together, what I'd like to show here is that biodiversity of terrestrial Singapore is predominantly insects, right? And of course, we want to think about the fact that all these things actually do count, right? So I was talking about how species matters. The second one, this is also not hot off the press yet. This is also a work in progress. This was only done in June this year. One of these pictures showed up in iNaturalist, right, by one of our macro photographers in Singapore. And I'm not sure you know, but we actually have a lot of macro photographers that are running around, that are taking very good pictures of the insects that we have here. Right, so this thing showed up. One of our collaborators said, this is a little bit odd, doesn't look like something that's familiar. Came down, and we went hunting, basically, right? And 2 weeks in, we couldn't find anything for the first 10 days or so. We got a bit worried. But at the end of the day, we're happy to share that we have found actually a new species of assassin bug, right? Right now, this thing is so rare that it's only endemic on one rotting tree log, right? Nowhere else. We've only found it on one tree log, and that's it. It's not even a fallen tree log. It's actually a standing tree log, right? So it's very particular. We're still looking at it. But beyond finding species, beyond finding out the names and describing them, they do interesting stuff, right? And this is actually one of the footage that we captured. It's actually a very unremarkable video footage, because all you're seeing is you see a dead ant that's being fed upon, or actually not a dead ant, this is a half-paralyzed ant that's being fed upon by the assassin bug. The interesting question here is, why is it already half dead when the bug actually hasn't even touched it, right? And this is something that we're still working on. Right, so in conclusion, right, the work continues. Right, we have a whole pipeline within the museum to try to churn out as much of these species discoveries as we can, right, with all the kind of documentation that is required nowadays, right, and along the way, try to have a good sense of what we have here. And of course, as mentioned, right, the insects that we have, please don't forget about them. Right. In fact, when we talk about conservation, when we talk about biodiversity, we do need to pay attention to them, because they do matter quite a bit. Right. Last but not least, right. Hopefully, you get to see that within such a small island that we have, we're actually sharing it with a lot of other species as well. Right. So as tight as it is, as packed as it is, we have to find a way. Right. With that, I'm out of time. Thank you very much, and I'm happy to take any questions.
So we'll now be opening the floor to any questions. I think we have time for one question. Does anyone have a question?
Hi, very interesting talk. How did you decide on the 3% threshold for the species separation?
Right. So the question is, how do you decide on the 3%, right? So the 3% genetic threshold is something that people actually have been using to estimate species. It's not a silver bullet. It doesn't solve all the problems, right? In fact, actually, I think Dr. Wendy Wang will talk about it as well. We do actually try different estimates. We do 4%, we look 3%, we look at 2%, and we look at the difference as well. But on an average, right, if you want to give a rough idea, for insects, especially in certain groups, 3% works relatively well. And that's why when we talk about the species, we actually talk about them as putative species. They're not 100% certain. Things can still shift around based on further work that's required, right? But we find it actually quite a nice handle to use.
Okay, thank you.
Thank you. Thank you very much.
Thank you, Dr. Hwang, for your talk. Our next speaker, Dr. J.C. Mendoza, is a senior curator of crustaceans and other marine arthropods. He is an active researcher on the systematics and taxonomy of true crabs, particularly of Southeast Asia and the greater Indo-Pacific region. He will cover some of the museum's marine biodiversity expeditions, from the initial organization through to the final discoveries. Let's welcome Dr. Mendoza on stage.
I'll use this mic. Good morning, everybody. So this is the title of my talk. We don't have much time, so I'll do this very quickly. So the introduction has been given. I just want to say I'm one of the academic offspring of our plenary speaker, and he's the one to blame for my experience in marine expeditions. Yeah. Okay, the seafloor, as you all know, is not flat, right? It's a multi-featured surface. And if you look at the average depth of our oceans, it's actually a staggering 3.7 kilometers, okay? Now, if you look at how much exploration we've been doing all this time, right? 200, 300 years exploring the sea and the seafloor. Most of what we've touched is actually in the shallow waters, okay? So the deep water has actually been quite relatively unknown, okay? In fact, we know much more about the surface features of the moon than we do our own planet's seafloor. Granted, the moon is much smaller than the Earth, but, you know, something so far away we know so much of, something just here in our planet we know little of, because there are a lot of challenges, okay? If you want a figure, we only know 1/5 of our planet's seafloor in great detail. Only 1/5 has been fully mapped, okay? And if we look at species, okay, the species that live in our oceans, right? There are many estimates out there. And of course, the consensus for all the species on the planet, okay, we have about 8 to 9 million species, all right? Estimated. Only 2 million of those have been described or are known to science, all right? And Dr. Hwang has already mentioned that insects are the stars when it comes to the proportion of species, all right? In the marine realm, okay, the situation is pretty much similar as well, okay? There's an estimated 1 million species known, 1 million species expected to live in the marine ecosystems, but only about 200,000+ have been described, okay? So in essence, we don't know 3/4 of all of biodiversity on the planet, okay? Both terrestrial and marine. So, in the past, people thought, we've been doing explorations for the last 300 or so years. There's very little to discover, okay? But they have been proven wrong, all right? So, this person, Professor Philippe Boucher, has been active in a lot of marine expeditions all over the world. He's a French scientist based in the Muséum National d'Histoire Naturelle in Paris. And he has led a lot of these expeditions, so he knows of what he speaks, right? And basically what he's saying is, we know very little, all right? And there's all of these things happening around the world. There's anxiety about sustainability and global environmental change, right? So us folks, we can capitalize on this and convince the powers that be to actually put more effort into funding and realizing exploratory expeditions. Okay? And where are these species likely to be found? Where? We are in the right place at the right time. Okay? In Southeast Asia is a transition zone. It's like the meeting place of two great oceans. Okay? And it has a very interesting geological history. Okay? So it's a very happening place. All right? And so this is one of the best places to be looking for species. All right? And Singapore, of course, is right smack in the middle of it. And this is why we have been sort of targeting places around the region. Okay? So there's been a history of expeditions around this region dating back to colonial times, right? And Indonesia in particular has seen a lot of this. But despite all of this, there's still a lot of gaps, okay? So we look at the history and then we see, okay, these places have not been visited yet, okay? And that's how we decide where to go next, okay? So the question was, okay, we've always been depending on developed countries to be leading these efforts, right? So some crazy people ask the question, can we do it ourselves? All right? And, well, I guess they said, yeah, let's try. Okay? And so it was settled that, NUS, together with some partners in Indonesia, would go and explore this area in the southwestern part of Java. Okay? And if you look at the bathymetry of the sea around this area, it's quite interesting because there's a gradual slope and then there's a very deep drop off. Okay? And depths here, for instance, can reach more than 7,000 meters. All right? So they started planning. Okay? First, of course, you have to do your site visits. You have to apply for permits, get funding. Okay? That one is vulgar to talk about money, right? So if you want to know how the money was sourced, Prof is still here, so you can go and catch him during the break, right? So look at this very organized course chart, okay? Planning is always like that, right? Oh, we have a nice, neat plan, okay? But the reality, of course, will prove different. So when the funding was confirmed, when the site was confirmed, the Indonesian partner was confirmed, then it was decided to call this next expedition the South Java Deep Sea Biodiversity Expedition, or otherwise known as the JADIS 2018. Okay? And the two main culprits, of course, are Professor Peter Ng and his Indonesian counterpart, Prof. Rahayu. Okay? And there were about 30 or so scientists who participated in this. Most of them, of course, came from the Tropical Marine Science Institute and the Lee Kong Chian Natural History Museum for the Singapore side. But we also had partners from Taiwan, from France, and of course, Indonesia. Okay? And the boat was provided by one of our institutional partners, the Indonesian Institute of Sciences, which is now called BRIN. Okay? And it so happened that around this time when these things were being organized, Singapore and Indonesia were celebrating 50 years of diplomatic relations. So the governments sort of capitalized on this cooperative activity as a further proof of cooperation between our two great nations. Okay? So this is what the course track actually looked like. Okay? So if you look at the yellow pins there, those are the places where the ship actually collected samples. Okay? So most of the collection focused on the Sunda Strait and the southwestern coast of Java. In the end, there were 63 stations in total, ranging from 100 to more than 2,000 meters in depth. All right? And all of these stations, of course, are geolocated, right? And all of this took place over 12 days. So we were stuck on the ship for 12 days. All right? And this is what the equipment, the main equipment that were used, okay? So two of these are like towed equipment. So they are being pulled along by the ship at a slow pace, and then it's being dragged along the sea floor. So we have a beam trawl for soft bottoms, and if you have a sea floor that is a bit more rough, okay, then we use the heavy-duty dredge. On the other side here, we have basically two pieces of equipment that specialize in grabbing or getting samples of the sediment in the sea floor itself, undisturbed, okay? So it can go down, and then it's brought up, and then the contents, of course, are analyzed systematically. So day zero, you can imagine we all fly to Jakarta, we go and meet our friends, our Indonesian friends, we go straight to the boat, right, and we start setting up. And there's a bit of work there, so folks like me who have not done an hour of carpentry in their lives are suddenly, pressed into doing it. All right? So there's a lot of flurry on the deck before we even set sail. And this is what the daily operations would look like. So the ship's crew, of course, will assist us in operating, in deploying and recovering the trawls and the dredges. Okay? So you can see here a dredge that is being brought up, and the contents are being emptied into these tubs here. Here is a trawl that has just been recovered. The cod end of the trawl is still down there, so it's being brought up. And this is what it looks like. It's full of mud, okay? So you can imagine there's some gems mixed in among that mud, okay? And here is a multicorer where each tube, you can see, has several inches of sediment as well as the overlaying layer of seawater. In the operations, of course, after these things are brought up, then we will have the difficult task of separating the specimens from the substrate. And that involves a lot of washing, sieving, okay? It's back-breaking work, okay? And we have to go through several trawls in one day. So you can imagine how busy it is on the deck of the ship in one 24-hour cycle, all right? But the end product of that, of course, is once you successfully finish washing your samples, then you have all those interesting things mixed up in there. And then you have to do sorting, okay? So I mentioned there were 30 or so scientists on board. Each of them have their specialties, right? So each of them will be targeting, of course, their own organisms of interest. So it becomes like a very busy marketplace. All right? So all of this happens outside the deck, day and night, right? Once we find what we're looking for, we quickly put that specimen in iced seawater and bring it back to the lab to be processed, okay? And what do? First, of course, we have to document the fresh condition of the animal. So photography is very important, right? And I'll tell you why. Because after you get the fresh specimen, you have to preserve it, right? And when you preserve something, after a few days, it won't look like what it looked like when it first came out of the sea. Imagine if it's in the museum for 100 years, it will definitely not look like what it did when it first came out of the sea. Okay? So aside from photography, of course, there will be an opportunity to get fresh tissue from the specimens, which can be used in future studies, right? Whether you want to do DNA or some special natural products, for example. Okay? After that, then we have to, prepare the specimen for preservation. All right? And some specimens require extra special care. They need to be relaxed before they're even preserved, either in alcohol or formalin. And while all of this is happening, of course, there will be daily, regular meetings, right? To react to what is happening in real time. Okay? You can plan and plan all you want, but there are always contingencies, or there are always events that do not go according to plan, right? So you have to do these meetings regularly so that you maximize the time and the effort while you're on the ship. It's not cheap to send a ship out there, okay? After that, okay, when we're done, okay, you look, we are very busy packing up, and you can see the relief of the participants in their bus back to the hotel, right? It's like, oh, we survived, we didn't sink, right? And there's this unenviable task of packing things and bringing them back to base, okay? So a lot of these things were sent back, shipped back to Singapore. So here's a picture of the truck delivering the specimens and the equipment a month later, okay? And we have to inventory it to make sure that things arrived in good shape, and then the museum research work begins, all right? And a lot of the work is actually done on board already. You have all the, the documentation that you can do. But even more, I would argue, is done when the specimens actually reach the museum. So this is where you do your real research. You compare the specimens with your reference collection. You start reading all the books and the journals that you need to in order to come up with a paper. All right? Aside from that, of course, the curator has to make sure that the specimens are in a good state and that they can be used by other researchers many, many, many years from now. All right? So let's take a quick look at the catch. Of course, we have a lot of deep-sea invertebrates. Okay? I won't belabor the IDs of these things. We have also a lot of deep-sea vertebrates, fishes especially. Right? And of course, I'm biased. I'm a carcinologist. So we had a lot of deep-sea crustaceans. All right? So you see every single photo here is based on the photos that were taken on board. That's why they're quite pretty, right? And the discoveries. Okay, so much has been said about species that have been described or genera that have been described as a result of this. So here is just a small portion of them. Okay, probably the most notorious is this deep-sea isopod that was collected during this cruise. But there are other groups as well: sponges, fishes, echinoderms, and other groups. Okay, so I talked about doing the work when we got back. So a lot of this is actually documented in a special issue that was published by the Raffles Bulletin. And it's free online. But if you'd like a hard copy, I think if you visit the booth, the Raffles Bulletin booth, you can still get some copies. Basically, the information is organized for scientists, but there are some papers mixed into this special issue that help summarize the whole effort and are very useful to share with members of the public who are not necessarily scientists. And of course, there's talks like this, right? So, very quickly now, this is my last slide. So, marine expeditions provide fresh impetus for biodiversity research. And they do this by, of course, filling specimen gaps in our natural history collections, such as our museums, providing fresh material for other studies, providing baseline information for marine biodiversity, which could be used for conservation, prioritization, and resource management, like fisheries, for example. Okay? And it's also very useful for promoting regional collaboration. And our relations with our Indonesian counterparts significantly improved when we did this, because now we met some of the young and up and coming scientists, aside from the veteran scientists. Okay? And of course, there are opportunities for providing science education, either by talks like this, workshops, interviews with the media, or special shows, right? So these are just some of the benefits that I can point out to you. All right? So I think I've run out of time. So thank you very much. This is just a picture of the team, the museum team that was on the ship. Okay? Oops. Right.
Okay, thank you, Dr. Mendoza. Since we are running a bit behind on time, we will not be doing Q&A right now. But if you do have questions, you are welcome to approach the speakers during our lunchtime. All right, so our next speaker, Dr. Wendy Wang, is a curator of insects and terrestrial arthropods, which are creatures that have jointed legs and an exoskeleton. Dealing with tiny arthropods such as ants naturally comes with sizable challenges, especially when sorting through your samples manually. Today, we can use DNA to sort our insect samples very efficiently, but there are some caveats which we will explore in her talk. Let's welcome Dr. Wang to the stage.
Okay, sorry about that. Okay, so in my work as a curator, and also for my research, I often have to process numerous samples of ants and other insects from biodiversity surveys and species discovery projects. So sorting all these samples to species visually is difficult and very time-consuming. Furthermore, for the ants, because of the existence of different castes, we get extreme variation between members of the same species. So for one colony, we can get queens, males, sometimes minor workers, and major workers. So all these look very different from each other, and it becomes a bigger problem if we have specimens in the sample that are not from the same colony. So one solution to this problem is to sort samples based on DNA barcoding. What exactly is DNA barcoding? The process can be broken down like this. First, we extract DNA from an organism. Then we select a target gene or part of a gene, and this would be our DNA barcode. Next, we create copies of the gene or the barcode by polymerase chain reactions, or PCRs. Finally, we read and compare barcodes between different individuals. So DNA for barcoding can be obtained from either of two parts of a typical animal cell. The nucleus, where you get nuclear DNA, and the mitochondria, where you get mitochondrial DNA. So mitochondria are organelles or powerhouses of the cell required for energy production and aerobic respiration. So perhaps the most popular DNA barcode is CO1. So this refers to the gene or part of the gene that codes for the protein cytochrome oxidase subunit 1. So CO1 is derived from mitochondrial DNA. Since each cell has multiple mitochondria, we can have many copies of CO1 per cell. So CO1 is present in all animals and plants. The main idea behind using DNA barcodes such as CO1 as a sorting tool is that individuals of the same species should share very similar or the same DNA. So even if they look very different from each other. So an example here is the driver ant. You see the sausage-like male versus the worker of the Dorylus levigatus. So take note that the DNA barcoding I'm talking about here is not meta-barcoding. Instead, we refer to individual-based barcoding. So these are two different concepts. For individual-based barcoding, it's like doing a blindfold taste test, where you try to identify different drinks by taking a sip of each separate drink. Meta-barcoding, in contrast, is like trying to identify different ingredients of a bowl of soup by taking a spoonful of that soup. So the ingredients that went into the soup no longer are in their original forms. So isn't it going to be too tedious to barcode every single specimen for very many specimens? To mitigate this problem, we have next generation sequencing, or NGS technology, which allows very many barcodes from multiple individuals to be read or sequenced all at once. So this works simply by allocating pairs of unique primer tags to each specimen being barcoded, such that the barcodes generated from each specimen by PCR will have a unique tag at each end. So these double tagged barcodes from different individuals can then be pooled and sequenced all together on an NGS platform. So the overall process is summarized in this infographic. At the end, after NGS sequencing, the DNA barcodes, which are double tagged, can be reassigned to their respective specimens of origin based on the initial assignment of unique primer tag pairs. So despite all its advantages, however, DNA barcoding is not perfect. So one problem we face is contamination. This affects up to about 5% of all sequences within a single sequencing run. So contamination can either arise from external sources such as DNA from indoor pests such as like houseflies or wood beetles. Cross-contamination can also occur between samples that are being prepared at the same time. So in most cases, we can usually detect obvious contamination by checking the identities of barcodes against public online databases such as GenBank, followed by cross-checks of morphology with the original specimens used to generate those barcodes. So another problem with DNA barcodes is that they may misrepresent the evolutionary relationships between species if they are analyzed for that purpose. So the processes of evolution are like long, complicated stories recorded in a thick storybook. So the thick storybook is like the total DNA makeup or the whole genome of an individual. So DNA barcodes are like the blurb or the excerpt to each storybook, a few short lines, very little information, and just enough to get a gist of the story. So when we try to compare individual DNA barcodes by constructing phylogenetic gene trees, we often don't get the whole complete story of evolutionary relationships between these individuals. So instead we get a rather unreliable, often poorly supported narrative of evolution. To make matters worse, unlike nuclear DNA, which is inherited by the child from both mother and father, so mitochondrial DNA, including CO1, is only passed down from mother to child. So by the same storybook analogy, this means not only do we not get the whole story from the short blurb that is CO1, we also get a one-sided, biased perspective of actual events in the evolution story. So usually for individuals of the same insect species, we expect CO1 variation to not exceed 3 to 4%. However, different populations of one species in different environments can develop very different CO1. So for ants, we have seen differences of up to 14%. So one example is when we compare geographically distant populations of the same species. So in this case here, for the ant, I named HPG species 7, based on nuclear DNA and morphology. These two ant colonies from Thailand and Lombok, Indonesia, actually belong to the same species, but their CO1 is more than 4% different, so about 5.4%. So this begs the greater question of at what percentage threshold of difference should we decide to separate species? Or how different should CO1 barcodes be for individuals to be considered distinct species? There is no universal percentage threshold for species membership or differentiation that applies to all species. So within species, differences in DNA barcodes can also be exaggerated by means of nuclear mitochondrial segments, or new mites. So new mites are fragments of mitochondrial DNA, which include CO1, that enter the cell nucleus and transfer to nuclear DNA. So a new mite copy of CO1 may mutate separately and become different from its original mitochondrial copy over time. So the problem here is that in DNA barcoding, sometimes both mitochondrial and new mite copies of CO1 can get sequenced. So when that happens, ants of the same species or even members of the same colony can have very different CO1. So I've observed up to 8% difference in CO1 between ants of the same colony, although they look exactly the same. So on the other hand, different species can have the same or very similar CO1 barcodes through interspecies hybridization or crossbreeding. So this applies especially to closely related species from the same locality. An example of this I came across in my research were these two ant species from one locality in Lombok, Indonesia. The two species are quite distinct from each other based on nuclear DNA and morphology. As you can see here, the one on the right is more elongate and slender than the other. So these two species are different, but they shared identical CO1, possibly from crossbreeding. So finally, bacterial symbiont infections can also affect species inferences based on DNA barcodes. So one prominent example is the endosymbiotic bacteria, Wolbachia, which infects roughly about 40% of all insects, including ants. So like mitochondrial DNA, Wolbachia is maternally transmitted. So in some cases, this bacteria helps to promote the survival of its host. Thus, infected ant queens may have higher rates of survival compared to uninfected queens. So a possible outcome of Wolbachia infection of ant queens is that different species of ants may end up sharing identical or similar CO1. So we are not sure of the exact mechanisms behind this, but some strains of Wolbachia tend to selectively infect hosts with a particular variant of CO1 that it favors. So let's say Wolbachia strain X favors the blue variant of CO1. So it infects ant queens of two species, A and B, with the same or similar blue CO1 genotype. So the two infected ant queens then outlive the rest of the uninfected queens and give rise to colonies all sharing the same blue CO1 variant. So in conclusion, while DNA is a useful tool for sorting and biodiversity discovery, we should always interpret results of barcode analysis with caution. So when it comes to clarifying species boundaries, it is always best to use DNA barcodes together with other forms of evidence, such as nuclear DNA or morphology. So that's the end of my talk. Thank you for your attention.
Thank you, Dr. Wang, for your talk. Our next speaker, Dr. Tan Heok Hui, is a senior collection manager of fishes. In his career, he has dedicated himself to studying the taxonomy, ecology, and biogeography of Southeast Asian freshwater fishes and has described over 100 new species. In his talk, he will recount the exciting discovery of an air-breathing catfish that was sighted for the first time in Singapore in 2022. Let's welcome Dr. Tan to the stage.
Good morning, ladies and gentlemen. We'll go straight into the talk, per se. It's okay. So, Encheloclarias, I've been thinking of the different ways how to present this, but just to drive a point home. The genus Encheloclarias, for most freshwater ichthyologists working in Sundalands, it's like the epitome of finding a dinosaur in your catch. It's like finding the freshwater coelacanth. I hope that drives home the point. It's not a common fish, and for the longest time, we have viewed it that it's a very uncommon, very unusual, and very, very difficult to get specimens of. Okay, and that's a dynamic photograph of one in the photo tank. This particular presentation is not possible for funding and a lot of support from various colleagues and donors. So Singapore's case, I shall start with this. It's very, very odd and very surprising. So first of all, this is a rough scheme of discovery of freshwater fish in Singapore's history. Started way back in 1849 and then right down to 61. Because post that, then there was the most comprehensive listing of freshwater fish of Singapore by Alfred. Okay, and none of them reported on this fish. None. So all the time, all this while, we thought, okay, that's it. We've more or less discovered what's there. So that's for the late Eric Alfred, whom we invited for the opening of the museum in 2015. He was the first one to compile all the freshwater fishes of Singapore and published in 1966. And I had the opportunity to actually ask him this question during an interview once, and I asked him pointedly, so, Mr. Alfred, when you do sampling of fishes in Singapore, was it exciting then, Was it the same as today? His response was, no lah, we just head north. Johor is much more fun. Singapore, you always catch the same few. So even back then, in the 60s, Singapore's freshwater fish scene was already depauperate. Okay, this was his map that illustrated Singapore in the mid-1960s, just to give you an indication how Singapore has changed geographically and the amount of reclaimed land that we have. And based on some studies that we did and published, Singapore actually has, on record, more introduced species than native species. Okay, and out of which 43 is the current number. Okay, never mind. And 22 of them are restricted to forest streams. Okay, so that indicates that about 75% of Singapore's fish are actually non-native. A small comparative study I did comparing with the freshwater fish fauna of Pulau Bintan, it shows you what we have in common with Bintan and what Bintan only has. This is just an indication of what Singapore's freshwater fish fauna could have been like. So, the case of Nee Soon. Swamp forest actually was fairly common in Singapore. But unfortunately, they were all cleared, partly drowned in reservoirs, partly because of industrialization. But fortunately, part of it was documented by Corner, but unfortunately, he was a botanist. So when he actually witnessed the clear felling of some of the swamp forests, he could see it, but he was only interested with the plants. So no aquatic organisms were collected. Okay, now this is, I would call this present day. Nee Soon swamp is the only remaining freshwater swamp left in Singapore. Okay, and I believe this was about 2022, I received an urgent message when I was overseas at that time. And I thought it was a prank message. No, it's not true. But they eventually convinced me otherwise. And unfortunately, they didn't have permits to collect it, so they released it. But we have followed them for the next round after explaining to NParks about the importance of this catch. And kudos to the students and research assistants that collected the fish. They took some photographs, even a short video clip, which confirms that this is not what they couldn't identify at that time. Okay, and to bring home the message, Alfred did not record the species. And when I interviewed some of the NParks people who had actually surveyed in the same areas, they themselves did not even catch the fish. So how was this fish obtained? Well, it goes to show that there are many ways to catch a fish. and we should apply these techniques. So they were using a baited trap which was left overnight, and that succeeded in getting this rather elusive fish. Okay, and now we know that this fish is around, and we have a rather healthy population as well. The history behind this is that it was only described in 1993 by Peter Ng and Kelvin Lim, and the original specimens were discovered in Pahang. There was a paratype from Kota Tinggi, and now it's in Singapore. And Singapore is the only place currently there's a living, healthy population in a protected area. There is another population in Pulau Bintan, but again, it's not protected. So in Singapore, we can say that we have a globally critically endangered fish. First vertebrate, I think. No, first aquatic vertebrate. So this shows a growth series. Okay, they're not easy to find. If you are doing active catching, you won't get them. So passive catching is one way. So this is in captivity. In fact, the same individual you're looking at is still alive. I've managed to keep it alive for like 2 years. They're not difficult animals to keep alive. You just got to feed them occasionally. Okay, and they are just like any other walking catfish in a sense. And this is from Bintan, with the opportunity of dissecting one as well, and realize that there's ripe ova, so that indicates that it doesn't have to be, it's not a very large growing species. Okay. A similar looking one from another island off Sumatra, but genetically rather different. Okay, I managed to get a small grant to do NGS on this little group of fishes. Results are pending, and we'll come to it. So this one, I've obtained 2 video footage from collaborators I get from the field. These are actually field hunters that get the fish. One in Bintan, one in Bangka.
This is how do we get the Encheloclarias fish.
So their way to get is very interesting. Anecdotal, and usually up to 5 to 10 individuals from each hole. And this one is in an oil palm plantation. See that at the end of the line, there's actually Encheloclarias.
You only get.
Okay. So the next few slides talks about the other species in this group of Encheloclarias. So the first one to be described was from Blaker, 1853. That's the plate that was reproduced, and this is the actual specimen. Okay, kindly photographed by the SIGNIFY team. And yes, so it's a very unusual catfish because it has an adipose fin, which is basically a fin that has no bones and it's filled with fat. Okay, so this is the discovery of it and the translation of description. So the original one from Bangka, and we managed to get fresh tissue. In case you're wondering why this one is missing a pelvic fin, because it's been clipped off for DNA studies. And the genus was actually first used in Harry and Myers in 1937. But they never stated why it was this name, but basically Anchylus in Greek means eel. Okay, and this is just an X-ray of showing that the species in Sundaland, the adipose fin, is not supported by bones, whereas the African species, which has an adipose fin, is supported by a bone. I mean the neurospines of the vertebra. And thanks to Kelvin, he made a cross-section of blackwater labyrinth fish, but as we now know from the anecdotal information, Encheloclarias can also be found in very shallow pools. That's confirmed by more and more substantiated in situ observations. Okay, and then this is the cornerstone publication by Peter Ng and Kelvin Lim, which till 1993, only one species of Encheloclarias was known to science. And in their revision, they actually described 4 new ones. Okay. So I'll just go through some of the species that they actually described from various places. And this fish was actually so scarce that many of the species were described only on one specimen. Okay. Unfortunately, some of them in a later publication been synonymized because we realized that they actually have many overlapping characters. So in the science of taxonomy, numbers do count. Your sample size do help to illustrate or support or to not support a certain conclusion. Okay, Curtisoma from North Selangor, Keloides, this is the one from Pahang, which is now found in Singapore. I shall give some clues to why and how I discovered it to be the same. So we had thoughts running through our head. Hey, is the Singapore one a different species? You know. So one of the tools that's very important, I just skipped through. Yes. So just to illustrate a point here, there was an ecological study by Thornton, based in Sabangau in central Kalimantan. They did catching of this fish in forested areas and in larger rivers. And their catch was quite interesting because it comprises of 27 species from about 4,000 specimens, and they say 8% of them were Encheloclarias. That's an impressive number. If I was on site, and as a practicing taxonomist, I would have kept every 300 of them. But unfortunately, they released all. Oh, that's fate. And in Brunei, the first discovery of Encheloclarias baculum. So this is the one that originally had two names, one from Sambas, one from Kuching, which later has been synonymized. Okay, and in Brunei, they're found in tip-up pools, which is caused by tree falls. Okay, this is a short video footage of why it's called the eel-tail catfish. Okay, this is the Brunei one, which is the longest amongst the Encheloclarias, longest bodied. Okay, that's a growth series. And if you think Encheloclarias baculum is elongate, that's not right. There are two other species of African clarids, which are even longer, and one even stranger, no pectoral and no pelvic fins. Okay, new range extensions. And so basically, it's a summary of what I've found so far. And what was very conclusive was the use of radiography. The number of anofin fin ray counts and vertebral counts can separate them into their own individual species. That's proven to be useful. But however, genetics tell a different story. And that's the next chapter. And this is the current distribution, as we understand it. They are closely associated with peat swamps, which are usually formed behind mangroves. Okay. Yeah. Thank you.
Thank you, Dr. Tan, for your talk. Our next speaker, Dr. Tan Ming Kai, is a curator of insects. His research focus is on Orthoptera, or more commonly known as grasshoppers, crickets, and katydids from Southeast Asia. In today's talk, he will share his experience collaborating with forestry departments and universities in Malaysia, Brunei, and the Philippines, and how these exchanges advanced biodiversity research in Southeast Asia. Let's welcome Dr. Tan to the stage.
Good afternoon, everyone. And today I'm going to share with you my experience with building collaboration and capacity while discovering new species of Orthoptera. So Orthoptera is an order of insects commonly known as the grasshoppers, crickets, and katydids. And what you may not know about Orthoptera includes they are more frequent flower visitors than we commonly thought, and some of them can even help with pollination. And Singapore recently has approved the human consumptions of insects, and a big proportion of them are actually orthopterans. And they can be considered healthier proteins with lower carbon footprints than conventional protein sources. Orthopterans can be found in a wide variety of habitats, including some of the less expected ones, such as sandy shores and mangroves. And the males of many orthopterans will produce sound to attract females of the same species, and this is called calling songs. And many of them are actually high frequency, can go up to 120 kilohertz. So there are approximately 30,000 species of orthopterans globally and more than 2,000 species in Southeast Asia. As you can see from this map of Southeast Asia, the discovery rate is not even throughout the region. And this suggests that perhaps the sampling has been fragmentary. At the same time, we've advanced in technology, greater ease to travel, access to information and resources, as well as collaboration. We have also found that new species discovery has been increasing exponentially recently. So the question that I do not know is, what are the other fascinating species out there waiting for discoveries? So for the last 16 years, I have been running around exploring, chasing, and discovering orthopterans from around the region. And at the same time, I also built ties with human beings. So I collaborate with university students as well as experts, and that include regional ones like University of Malaya, University of Brunei Darussalam. I have also worked very closely with forestry departments such as the Forest Research Center in Sepilok, Sabah, and they have provided a lot of helpful support in terms of manpower as well as logistics. And of course, there are also other scientific research institutes such as the Sakarat Environmental Research Station in Thailand. So collaborations are essential. We always need to apply for permits, be it research, collection, or export permits. And we need the support from these local collaborators. And these local collaborators are also the ones who probably know more about the procedures involved with such application. An example that I want to share is that in Malaysia, applying for a research permit in Sabah is very different from the procedure in Peninsular Malaysia. Occasionally, we also need to seek permission from local forestry officers before we enter the forest. We also seek permission from indigenous people, which I will talk about later. And lastly, we also sometimes need to inform military groups, especially in places which are politically less stable. So we need to inform both the government as well as the rebel groups to make sure that we are not enemies by any of one of them. So in essence, collaboration is also essential for safety. And one of such places is Mindanao in Philippines. But Mindanao is also a super diverse island in the Philippines. And I had the privilege to work with a few PhD students and master students from the Mindanao State University. So to me, it's actually a two-way transfer of knowledge. So while I provide the taxonomical and entomological guidance for these students, their collection actually helps to expand and advance my knowledge and experience about the species that can be found there. So in other words, such collaboration help to expedite discoveries. So for the past 2 to 3 years, I have students who have found new locality records of crickets in the Philippines, and of course numerous new species of crickets and katydids from different parts of the island as well by different students. I have also students who record the calling songs for orthopterans, which are always very important and precious data. So collaboration also opened up horizons. So here I want to specially mention Tony Wabilat from the Paris Natural History Museum. So I have known him since 2011, and for the past 15 years, I have learned and observed a lot about him. About taxonomy, both in the lab as well as in the field. And I guess since then, we have been close collaborators, and he is a mentor for me. But I think we have also developed to become close friends and confident. Another example, another collaborator I would like to mention is Fernando from the University of Lincoln. So prior to meeting him, I focused solely on the morphology, but he widens my horizon by showing me how to record and analyze the calling songs of orthopteran, especially the ones that produce ultrasound. So here is the first paper that we co-authored. So we recorded the calling songs of 6 species of Arthropodes katydids. All of them are produce extreme ultrasound, so completely inaudible to us. But when we analyze the calling songs, we found that the core structures are very different between different species, both in terms of the temporal as well as the frequency spectrum. We also examine the stridulatory apparatus or organs that are responsible for producing the sound. And as you can see, they are also very different between different species. So by integrating both morphology as well as the calling songs, we are essentially trying to do what we call integrative taxonomy. So lastly, I would like to mention about the indigenous people. So indigenous people is a little bit foreign concept for Singaporeans, but these are people who live intimately and closely with the forest and in the nature, and they are very much fine-tuned to the local environment. So whenever I'm out in the field, I often have to rely on them bringing me around in the forest as well as them taking care of me. And of course, not forgetting the animals that they also bring along to help us. However, at the same time, they are also often marginalized and taken for granted in the academia. So for my part, I always try to make sure that I can at least write down their name in the acknowledgement segments of the paper. But I think there's also other ways in which we can recognize indigenous people. So this includes naming species after indigenous people. So this is an example, Lumat. Lumat is referring to indigenous people in southern Philippines. And the word itself means indigenous in Cebuana language. And the inspirations come from our fieldwork in Siargao Island in the Philippines. The car broke down in the middle of nowhere. So while we were waiting for our support and help, I got a crash course on the Philippines history by my collaborator's daughter, who at that time was very passionate about history. So I have also described species named after, with a name Kenuan. Kenuan means for you in Iban language, and Iban language is spoken by Dayaks, or a group of native people in Borneo. There's also another cricket, Nicitrus rindu. So this species was described in 2021. It means love in Iban language, and in Bahasa Melayu, it can also mean homesick and love, missing. So this comes about because during those times, many people were separated from their loved ones because of lockdowns. There are people who lost their loved ones. So this inspired us to come up with this name. So in conclusions, in science, we try to grasp the truth in an objective manner in the pursuit of scientific knowledge. And this is the same for taxonomy and taxonomists like me. Taxonomy is a science. But to me, the opportunity to be able to live, interact, and learn from people from different walks of life in nature is also very humanizing and humbling experience. So this completes the human experience. And with that, thank you for your attention and a few of my favorite discoveries. Thank you.
Thank you, Dr. Tan. Our last speaker for the session, Dr. Marcus Chua, is the curator of mammals at the museum. He is interested in Southeast Asian mammal evolutionary history and the ecology and conservation of mammals in threatened landscapes. His work on the critically endangered Sunda pangolin in Singapore highlights how citizen science and data collection are key to conserving threatened species. Let's welcome Dr. Chua to the stage.
Thanks, Mavis. Hi, everyone. I'm Marcus, the curator for mammals at the museum. I'm the only one coming in between you and lunch, so I'll be quick. I don't have great expedition stories or new species to talk about, but I have an animal that would steal your heart. And my talk is about putting this species, the Sunda pangolin, in Singapore on the map for conservation. So what is the Sunda pangolin? So this is a species of mammal that's unlike most cute, furry, cuddly, soft animal because they're covered with scales. They're closely related to carnivorans, dogs, cats, but they belong to the group by itself. Because they eat ants, they are also known as scaly anteaters. The scales on their body are similar to the material in skin, hair, and nails. And because of that, they are caught for their scales for traditional medicine and for meat. But they're actually really shy. They come out at night, and they are really hard to study. But because of they are hunted, they have the dubious honor of being the world's most trafficked mammal species. And they are listed as critically endangered worldwide, both for the Sunda pangolin and many pangolin species. In Singapore, hunting is not so much of an issue, but they are threatened by habitat fragmentation, loss, as well as they fall victims to road traffic accidents and roadkill. This is an example of one that's reported in the press. They are in such poor state that experts around the world have come to Singapore in 2017 to decide on come up with a plan for the conservation for the world's pangolins, and at that same time, a plan for Singapore's pangolins too. And this was done at Mandai Nature, and we come up with a plan, Singapore Pangolin National Conservation Strategy and Action Plan. At that time, we realized that we know so little about pangolins that we did not have enough information for their conservation. So one of the first goal of this plan is actually to gather and share information for pangolin status, ecology, biology, behavior through studies. And some of this come from the specimens that we collect from roadkills that you saw in the past. So that becomes our specimen, and we learn from them. And one of the most important things is to understand the distribution of pangolins in Singapore so that we know where they are, we can better protect them. But although this plan was published in 2018, there was very few studies done since then, and this was an unknown, this remained an unknown information for many years. Until the COVID-19 pandemic happened, and we all stuck at home, and we have little time to, we had some time to reflect on what we need to be done. And at that time, the lead instigator, Bichu Strange, from the Nature Society of Singapore, said that, hey, let's do something for pangolins. The Nature Society has some money from donations. What can we do? So I thought about it, looked at the Conservation Action Plan, and said, let's try to map pangolins in Singapore to understand more about their behavior, their distribution. And when it comes to maps, we need a GIS expert. We worked with GIS consultant Tony O'Dempsey from Nature Society. But we need someone also who's able to devote time on this project. And so we looked, someone who's actually very detailed and can actually deal with all the information. Zoologists are not very useful, according to Wei Song, for that. So we looked to botanists. So there was a graduate from NUS Department of Biological Sciences who worked with Guat Yen at NParks, who was in between work, and we had interviews, and we found that he was the most suitable candidate, and he hired him. So how do we deal with pangolin records all around Singapore to map them? So I had some experience supervising a student with Sivas from Department of Biological Sciences, and we waded into roadkills in Singapore. And this student, Ong Shu Yi, realized that the information in Singapore for roadkills is extremely fragmented. It could be from Nature Society, from the museum itself, information from NParks, and from ACRES, the rescue organization in Singapore, and it's a wealth of information to go through. So with that, we decided to target databases which had the most information, and this was from the Singapore Pangolin Working Group, which has been acting to aggregate the information if you report roadkills to them. ACRES, Nature Society of Singapore, the museum's own records, as well from specimens, as well as from public records that have been reported to us. From Mandai Nature, specimen animals that have been rescued goes to Mandai Nature. They get information too. And we also scrub through social media posts on Facebook. And with that information, we divided that into roadkill animals that are dead, rescued animals that were brought to, that rescued alive, as well as sightings from members of the public, and they all considered our data. We had to do extensive data quality checks to remove duplicates, remove records that we were unsure of. They either lacked location descriptions or did not have enough clarity. And with all that information, we conducted this exercise called georeferencing. It's a science in itself. We followed best practices that were published by experts. And with this information from the database, we could do analysis and visualize the information. And most of this information were actually from public records. So what did we find? So this is the map of pangolin records in Singapore from 1996 to 2021. There were 482 records. And the location of these records tell us that most of the pangolin records were found around core nature forest areas in the Central Catchment Nature Reserve and Bukit Timah Nature Reserve and the Western Catchment. The spots in magenta or red are roadkill. Those in blue are rescues. Yellows are sightings. And although most of these records are around close to nature reserves and forests, there are also records away from this area, such as in Joo Chiat and Katong area, in Telok Blangah, Sentosa. So how do pangolins get from the core forested areas to these areas? So this remains a question, and ACRES CEO Kalai thinks that they might be using the longkangs, the drains in Singapore, as a network to move around. But you go around from one side, it could be forest, you come out the other side, it's housing estate, so they need to be rescued to be brought back. We are also extremely lucky because you are here in Utown, NUS, where a pangolin has been rescued before. But what else did this tell us? So there is evidence that there's an increase or more records now for roadkill rescues and sightings for pangolins compared to 10 years ago. But there is strong evidence for the rise in roadkill reports since from 2011 to 2020. So this is a worrying trend. And also, we could tell who is most at risk from the data. So based on the rescues from 2011 to from 1996 to 2021, male pangolins were 2 times as likely to be rescued compared to females. And this is reflected in roadkill records where male pangolins were 3.6 times more, there were 3.6 times more roadkill of male pangolins compared to females. And this is consistent with their behavior for other pangolin species where it seems that males and sub-adults, like not fully grown adults, tend to be moving around a lot more, and they cover a lot more distance in one night, up to 7 kilometers in one night, possibly to disperse, to find new territories, or to find mates. So what can we do? What do we do with this information? So the fact that pangolins are found mostly around the forested areas suggests that there are hotspot areas where mitigation actions should be focused. And just earlier this year, before the general elections, it was announced that there will be fences that will be built around the nature reserves to prevent roadkill. So the pangolin is one species that can benefit from this sort of conservation and management effort. 2, we learned that sub-adult as well as adult males are most at risk from roadkills as well as being for rescues. So it informs threat mitigation as well as post-release strategies. When pangolins are rescued, this information would suggest that it's best that we release them far away from the roads, as close in the core center part of the forest as possible, to prevent them from coming out and put themselves in peril. So this is an example of pangolins being rescued. But there are some positive findings as well. So this is the first georeference database for pangolins anywhere in the world. And this is the starting point for further studies for the species in Singapore. And since 2015 to 2021, there has been a standard operating procedure for pangolin rescues and roadkill. So this requires close coordination between members of the public that reports, wildlife rescue as well, or NParks that picks up the pangolins. They are all live pangolins that are rescued are sent to Mandai, where the vets will examine them to learn more about them, find out what's wrong with them, and they get treated for any injuries if they are injured. And once they're given a clean bill of health, they are released again. And it's been reported that 67% of all rescued pangolins are successfully treated and returned to the wild. And from this, the vets get a better understanding of pangolin health in Singapore. And all this requires the strengthening of this network because it will be crucial for the conservation of pangolins in Singapore. So this information has been published in the journal Oryx, and I'm happy to report that all major members of the Singapore Pangolin Working Group and Parks Nature Society of Singapore, NUS are involved in this study. If you want to read the paper, you can scan the QR code. But there are actually real-world conservation applications that come out of this. So the database has already been put to use for pangolin conservation. Since its publication, it's been used in 9 development projects to inform developers of pangolins in the area. So we typically get an email from a developer like this saying that, oh, we've been referred to your group by NParks. Could we get information about pangolins in this area? The thing about pangolins is they're actually extremely shy. So within the short environmental impact assessment study period, some of them do not get detected, even though if they are in the area, they use the area. So this database actually is very useful for to inform developers that, hey, there are pangolins in this area, you want to watch out and be careful with mitigation or planning how you build the areas. So all this would not be possible without the close work. This is the best group project I've worked with multiple partners. With ACRES, Mandai, Wildlife Group, and Parks as well as Nature Society of Singapore, and people that have been maintaining the database and records before me, Calvin Lim from the museum, Paige Lee and Francis Look from the Pangolin Working Group, and everyone who has contributed to the Sunda Pangolin record. Your work has come into this database as well. And also there are heartwarming stories where during the pandemic, this is a teacher who has been conducting safe pangolin conservation dinner at home, where she would have different courses of dishes talking about pangolins. This is a mud pie with termites sticking all over it, talking about how pangolins eat. And the proceeds actually got donated to this project. And if you were using your DBS banking app during that period, you'll notice that DBS Leaf Well had a pangolin fundraising effort that also contributed to this project. So this was really a heartwarming story for pangolin conservation. Well, this is the end of my talk. Thank you very much. If there are time, we'll have questions. If not, look for me after that. Thank you.
Thank you, Dr. Chua, for your talk. Before we break for lunch, we'd like to take the opportunity to introduce our new exhibition with the Science Center called Dinosaurs, Extinctions, and Us, which will be held from October to March. If you're interested, we will be playing the introductory video during the lunch break, and you can scan the QR code for more information. We'll also be playing Science Stand, our YouTube series, where you might see some familiar faces. Lastly, before you go for lunch, we'd like to take a quick photo. So if you guys on the side could please move into the center of the room, that would be really helpful. We will just be doing a quick photo with everybody here today.
Okay, good afternoon, everyone. I'm Noman from the Outreach and Education team, and I will be your MC for this afternoon session titled Specimens to Stories. Before we start, a gentle reminder that strictly no food is allowed in the auditorium. Videography and flash photography are also not allowed. In this session, we will explore some ways the museum is bridging the gap between science and the public. This can come in the form of stories, media, and many more. As with the morning session, each speaker will present for about 12 minutes, followed by a short Q&A session right after their talk. If you do have questions, kindly raise your hands and we will pass a mic down to you. Before we begin with our first speaker of the session, I'd like to mention that there will be a panelist session at about 4 p.m. Whatever burning questions you have, do submit them by scanning the QR code shown here. The QR code will also be shown during the tea break at about 3:45 p.m. For our first speaker of the afternoon, we have Miss Kathy Poh. Miss Poh is part of the museum's Biodiversity Histories team, where she leads an oral project about the museum's history and conducts research on Singapore's past biodiversity. Now, when you look at zoological specimens, have you ever wondered about the stories behind the people who collected them? In her talk, Miss Poh will be sharing about the work of Guy Charles Maddock, an ornithologist who published a book on birds while interned in Changi prison under the Japanese occupation. Let us welcome Miss Poh to the stage.
Is there a handheld mic? Sorry.
Okay, hi everyone. I know it's post-lunchtime, so I'll try not to make this a bedtime story. So today I'll be talking about this book, by Guy Charles Maddock, and we'll be looking at the stories behind Maddock himself, as well as the making of the book, and more. So before we get into it, I wanted to talk a bit about what we do in the Biodiversity Histories unit in the museum. So I do oral history, and I'm also, I guess you could consider my work to be that of an archivist, because for us, we collect and curate historical materials relating to the museum and also the collections that we have. So this could be things like 35mm photographic slides, or photographs, or field notebooks, and things like that. And these are some of the books that we have published in recent years. So one that I'd like to point out is Singapore at the Dead Zoo, which is by our SIGNIFY team, which will be explained more by Tricia after me. So this is like, I guess, quite a broad overview of the kind of materials we work with. So our museum, our collections, and also the broader past of Singapore's biodiversity. And I would like to start my story today with our museum gallery. So how many of you all have seen this in our museum? Okay, so quite a lot of people. For those who haven't seen it yet, maybe you can drop by sometime soon. But so basically what we have here is this display case that talks about science work that was done in Singapore during the wartime years. And the materials pretty much focus on this guy named Guy Charles Maddock. And we've got his specimens, his specimens, and also some materials here, some paper materials. And actually I would like to bring our focus to this flash of red over here, which is the book titled An Introduction to Malayan Birds. And as you can see from the pages here, I'm not sure how clear it is, but this book was printed and published in Changi prison. And the date written there is May 1943. It's one copy only. And there's a bit of an excerpt from the preface over there. But so after the war, this book was also revised and republished in multiple editions. And now it's recognized as one of the early works that of significance about birds in our region. But I think for me, as someone who does history, what is more fascinating in this case are the human stories around the book and the conditions that it was created in. So just a short introduction to Maddock first. So he grew up in the Isle of Man in the United Kingdom. And in 1931, he came to Malaya by ship from London. And he was joining the colonial Malayan police. But in his free time, he was an amateur ornithologist, and this was an interest that he got from his father, who worked on birds in the Isle of Man. And when World War II began, Maddock was interned at Changi prison, and in the later part of the war, the prisoners were also moved to Sime Road camp. And this is the period of time that the book was produced and used in prison. And also, after the war, Maddock spent some time in Singapore before going back to the Isle of Man for his retirement. And actually, with Maddock's story, not that much has been known until the recent few years. And a lot of this is with thanks to Fenella Maddock Davis, who is his daughter. And as you can see, so I was able to visit Fenella in her home in the UK maybe 2 years back, and she spoke to me a lot and told me a lot of stories and also showed me some artifacts. So for example, we have in her photo album, there's a photo of her in the museum before it opened in 2014, and we have Dr. Tan Swie Hee there as well. And also, apart from sharing her stories about her father during the war, she also showed me this monocular, which is basically a binocular but with one lens. And she said that it was something that he had with him while he was in prison. And so now I will move on to talking more about the different stories that, different snippets of stories that she's talked to me about during the time that I spoke to her. And I think one of the things I wanted to start with was with the making of the book, because, I mean, even now, if you ask me to make a book like that, I don't think I could do it, much less thinking about how it was possible during the war. So with this book, the paper was actually stolen paper from the Japanese commandant's office. So one of the prisoners, I think, was on good terms with someone, and then they were able to get the papers from the office. And what you see here, the red material covering the book, it's said to be leather red, which is kind of like a synthetic leather. And according to Fenella, she was saying that during the war, the civilian internees were also brought into the city to clear up debris from, for example, like bombings, and these sorts of things. And this was also a chance for them to salvage materials. So someone could have been looking for the leatherette. So this would have come from maybe like the seat cover of a car. And also the materials used to do watercolor would also have been procured in the same way, probably. And these drawings were done by David Molesworth, who was a doctor in Malaya, and he also has a very deep interest in ornithology. And these drawings would likely have been done from memory as well. And so the text is typewritten. I think across the whole book, the depth of the ink is quite different across the pages, and this is because there was only one typewriter ribbon. So what the prisoners would have to do when they were typing it up was that they would have to flip the ribbon over and over. Oh my God, I am going too slow. Hold on. And so basically this was how the book was done. And I will share more stories now. So I think the other quite amazing thing for me when I was listening to Fenella tell me about the book was how it was actually used during the war. So it was not something that, was made just for fun, but it was passed around in the prison. And on one occasion, Maddock himself also spoke to a group from the men's camp to share about ornithology. And they also managed to pass the book over into the women's camp, and Maddock himself was also asked to go over to the women's camp to give a talk. So this was a time when I guess the policing of the camps was a lot more lenient, but this was quite a short-lived thing, because I think in October 1943, so a few months after the book was made, the Japanese became a lot more harsher with the way that they ruled the prisoners. So a lot of this became impossible. And I think apart from stories about the wartime, I also got to hear about Maddock's book, Maddock's work before the war as well. And I think one of the things that I would like to highlight was the role of this person named Majid, who Fenella described as a handyman, but we might consider more of a collaborator or a collector who worked with Maddock. So I think what was interesting here was that she said that Maddock and Majid both respected each other hugely, and they were on an equal footing. And Majid was also someone who was doing a lot of the active work of collecting and even the constructing of bird hides, which you can see here. And also you can see these photos of Majid with Maddock, and this is David Molesworth. So they were working quite closely, and these photos were also generously shared to me by Fenella as well. And I think the last major point I wanted to touch on was a personal significance of the book. So actually the book itself, until now, it's still within the possession of Fenella's family. And this was something that was not originally intended at first, because Guy Maddock wanted to donate all his materials to the Natural History Museum. But Fenella said that when her father showed her the book and was, explaining his will to her, she was like, actually, I think we should keep it within the family, because this is an heirloom, and this is something that is very important for us to keep and remember. And because of that, Maddock, according to Fenella, reluctantly altered his will to keep the book within the family. And it's still a book that she brings out and show to her grandchildren, and they talk about the book quite regularly. And I think even when I visited, she was also very generous in allowing me to handle the book. So I think with being a museum, often we think about all things as very, like sacred and very precious and something that maybe we shouldn't handle. But in this case, because it's something that is also an object that is very closely tied to the life of Fenella and her father and the rest of her family, it's something that I think takes on a different meaning. And this is something that I felt, it was something that moved me a lot in my conversations with Fenella. And she also shared some photographs of her father, and I think these were taken after the war. So Fenella also told me about how she went out with her father to places like Fraser's Hill in Malaysia to look for birds' nests. And so I don't think I have a lot of time left, but I'll just speed through the remaining slides. So these were photos from Fenella's visit to Singapore in January. And this was also the time when she came over to allow us to digitize the book that her father wrote and produced, and as well as some photographs like the ones we just saw in the previous slide. And during the week, we also brought her to our museum to look at birds in our collection, which Yenny hosted very kindly. And we also held a panel session bringing in Keita from Bird Society, Anthony Medrano, who used to be in the history department. And it was a lot of fun, I think. And these are also some photos from Maddox's own birding trips. And I think it's quite interesting because, photography was still very expensive during the 1950s. So this is material that really captures like a specific time period that we can't really go back to now. And that's all I have. Thank you.
Thank you for the sharing, Ms. Poh. Right now, we will be opening the floor to any questions you may have. If you have any questions, you can raise your hand. Questions? No? Okay, I have a question for you, Ms. Poh. So what is the most memorable thing about this project?
The most memorable thing about this project? I think it was... I think really it was like during the interview sessions, hearing about how much the book meant to Fenella. Because also the thing about all these stories, I think I was also asking Fenella, like, why does she, or like, how did she get into doing all of this research? Because actually her father was, according to her, he didn't talk a lot about his life during the war because it was something that was also quite traumatic as well. So a lot of these stories were only collected from people around him later on in his life, or even after his death. So I think for me as an interviewer and someone speaking with her to collect information in this way, I don't really know how to put it also, but I think it's the fact that, like these stories, they are not just facts of history, but very much lived experiences as well.
Thank you. Any questions from... okay, we have one hand there. Okay.
Yes. So the book has been digitized. I forgot to mention. So the book itself and the photographs, such as these ones, they are available on NUS Library's website, Digital Gems. So I think if you Google Maddock Introduction to Malayan Birds or Guy Maddock Private Papers Collection, so that line there, sorry it's a bit small, but these should all be public access. So you'll all be able to look at the book, like the full book, with all the drawings and illustrations and text, as well as the photographs. And all of this is online. Was this the first book on Malayan birds? No. I don't think... No, there were earlier books also by people like Claus and Chasen. So this was not the first, but it's one of the early works. No problem.
Thank you for the sharing, Ms. Poh. Our next speaker we have today is Ms. Tricia Cho. Ms. Cho is a research assistant for a museum project named SIGNIFY, which stands for Singapore in Global Natural History Museum's Information Facility. Her work involves managing Project SIGNIFY's database and social media content. For more than 2 centuries, naturalists from around the world have collected specimens from Singapore and its waters, leading to local specimens being housed in natural history museums worldwide. SIGNIFY, which began in 2022, aims to trace the footsteps of these specimens through digitizing and documenting them. Let's welcome Ms. Cho to the stage.
Okay, hi everybody, I'm Tricia. I've already been introduced, so I'll just continue. Yeah, so today I'll be sharing the story of SIGNIFY and telling all of you a bit more about how we transform specimens that we see into images. So this is a bit of what I'll be covering today. And before that, I'd like to give a bit of context for SIGNIFY. So this is what it stands for. And basically what the project does is that we go to natural history museums overseas and we digitize all the specimens that were previously collected from Singapore. So this is a donor-funded project and it's a 5-year initiative of the museum. The project itself has 4 main aims, right? So the first, of course, is to image and document scientifically and historically important specimens. So what we mean by this, scientifically important specimens are those like important type specimens and reference specimens, and historically important specimens are those that were collected before 1920s. So the second point is to provide Singapore context. So these are things like interesting specimen stories and collector stories, which I'll be sharing a bit more later. The third is to provide research-ready resolution images. So these are very sharp images that allow us to refer to the specimens even though they are housed overseas. And lastly, of course, in the process, to facilitate bilateral research. Okay, so now let's go back to the story of SIGNIFY. And as I mentioned earlier, this is what SIGNIFY does. And I guess the next question is, why are Singapore specimens not in Singapore, right? Why are they found overseas? So this goes back a bit into the history of Singapore. About 200 years ago, with the founding of modern Singapore, and Singapore was developed into a successful trading port. And with that, we have a lot of travel in and out of Singapore. And we had a lot of collectors visiting Singapore as well. And from there, they collect the specimens and they bring it home. Right? So after they bring it home, they do one of 2 things. Either they keep it in their private collections, or they sell it to museums overseas to earn a quick cash. So from private collections, a lot of these are eventually donated to the museums after their passing. Right? So this is how our Singapore specimens are being found overseas. And actually in Singapore, we have a very large number of collectors that are being recorded in the database. So here is just 250 as an example, and today I'll be covering 2 famous collectors as an example. So the first is Sir Stamford Raffles. I'm sure most of you have heard of him, the founder of modern Singapore. And apart from his political influence, which most of us know about, he was actually very interested in collecting specimens and natural artifacts. And I think we're actually very fortunate to still have some of his specimens being around and being digitized, because on his way back to the United Kingdom, the ship that he was on actually sank, caught fire and sank, and with that he lost a very big collection of his papers and specimens. Yeah, so here I would like to give 2 examples of the specimens that were collected by Raffles. So the first one is the Raffles Banded Langur, was actually named after Raffles himself. So this was actually one of the first specimens that the team imaged when we went to London in 2022. And this was the first ever primate that was recorded from Singapore. And second is the green broadbill. So this too was one of the first specimens that was being imaged when the team visited London. And this specimen that was collected by Raffles was actually named and described by Raffles himself. Right. The second famous collector you heard of is Wallace. It's also a very known explorer and naturalist, and he was known for his developing the idea of evolution by natural selection, independent of Charles Darwin. So Wallace was actually, he explored the region quite a lot, so he spent a total of around 8 years in Southeast Asia, and within the 8 years, he spent about 228 days in Singapore. So Wallace was actually quite a prolific collector. Yeah, he collected thousands of specimens, but here I'll just highlight something interesting about him. So this is a beetle specimen that was collected by Wallace. And something interesting about Wallace is that he's actually a very conscientious person. So he will always label his specimens, and if we look specifically at insect specimens, he would always include a round label. It's around 8 millimeters in diameter. And on the round label, he would include abbreviation of the location where he collected it from. So for Singapore specimens, you will see a short form SING on it. And this is actually quite a telling characteristic. So if we see any specimens with this round label, it's most likely collected by Wallace. So going back to SIGNIFY, the project was officially launched around 3 years ago, and this was a bilateral event. So we had it here at the museum lobby and concurrently in London. And as you can see, we were waving at them through a Zoom call. Right, so London is one of our first partners because of our shared history, and because of that, we have a very large number of specimens that's being housed there. So now I think this is the interesting part of the talk, which is how do we image our specimens and turn them into images. So we start off, of course, looking into published literature and museum databases. And the next thing is when we get to the specific museums, we carry out physical search. So we physically look through specimens and written ledgers, looking for the word Singapore. So it's over here. And this is very important because a lot of times information is scarce, and even mining all the literature and museum databases will leave out a lot of specimens. And this is the equipment that we use to digitize our specimens. We suggest some of them, and it's very important for our equipment to be portable, right? Because SIGNIFY is one of the first mobile digitization units. And what this means is we travel with all our equipment, right? So everything has to fit within the luggage, and we're often constrained by the weight that's allowed by the airline. So it ranges from 23 kg to 30 kg per luggage. Yeah, and it's important for our equipment to be versatile as well. So we have to image slightly smaller specimens or larger bird specimens as well. And for our equipment, we control most of it through our laptop, and this connected to our main setup over here. So this is to prevent shaking of the camera when we image our specimens. And because specimens are three-dimensional and not flat, we do something called focus stacking. So if you can see, we actually image at different heights so we can get different focal points. And then we'll stack the image together using a software, and then we'll get a very sharp and clear image of this beetle here. Yeah. So this is how we get the image, and after that we'll have to do post-processing. So this is my colleague using Photoshop. She's picking out the flying fox specimen here and erasing the background. Right? Yeah. So something like that, and this will have to be repeated for all the different views of the specimen as well as the label. So eventually you get a collage of images, and this is what we call a figure plate. And what we also call is research-ready resolution because it's very sharp. So here I'll give you another example of the red-crowned barbet, important type specimen that was collected. So this is actually the bottom view of the bird. And as you can see, it's very sharp. You can see all the fine feathers of the bird. Right, so that's how we image our specimens, and now I'll move on to some of the outputs by the project. Yeah, so the website is our main data portal that provides anyone, anywhere, and anytime access. And from our website, you can access all our figure plates as well as the associated data. So these are things that we could have found from published literature or on the specimen labels itself. And you also have some of our project publications as well as interesting specimen and collector stories. So these stories is done in conjunction usually with our social media. So these are some examples here, but this is also similar to the Wallace beetle example that I gave earlier. That was just a snapshot of our social media. Right. And also, apart from that, we of course have collaborations with our partner museums. So this is one example with the National Museum of Ireland, where we did this book called Singapore at the Dead Zoo, which Kathy mentioned earlier. So this was actually funded by the Embassy of Ireland to Singapore to celebrate 50 years of diplomatic ties. So this book features all the interesting specimens that the team imaged when we were there, and this is an example of the story. Yeah, so this is a collection of Armstrong's molars, and it actually states how it was donated by Miss Elizabeth Armstrong, which is his sister after he passed. So if you're interested, go and read the book. It's on our website. Yeah, and I think apart from the scientific community, we also want our images to be utilized by anyone that is interested in natural history, right? Not just scientists. So earlier this year, SIGNIFY was invited as an industry partner by LaSalle for their design communications module. So the students were tasked to use specimen images and create, I think, like a informative output. So it can be soft copy or hard copy. And these were some of the interesting things that they came up with, which I think was quite nice. And with that, I come to the end of my presentation. I'd like to acknowledge donors, partner institutions, and my colleagues. Thank you.
Thank you, Ms. Cho. Okay, we will now open the floor to any questions you may have. Okay, we have a hand over there at the left.
Thank you for sharing, ma'am. From my understanding, I believe there's a website called like the Biodiversity of Singapore, which also has like similar images shown online. May I ask like what are like the differences between this project and that website, and if there's like any like if this is meant to be like an overhaul of that website or something?
Okay, thank you. For Biodiversity of Singapore, it's the images are also featuring Singapore specimens. A lot of them within the museum itself, so some of them are our museum specimens. But for SIGNIFY, we do travel overseas to image the specimens that are housed in the overseas museums. Thank you. Yes. On the labels, it was recorded to be collected by Raffles, but we'll never know if, like, you know, if you went as a group, whether it was collected by someone else then as well. Yeah. How many more? As many as we can. Yeah, so I think initially we had a target of 10,000, but to be honest, I think it's a bit unrealistic. So currently, we have 3.7 thousand that are already imaged. The team is currently traveling, to image more specimens. And so as much as money and time permits, Thank you.
Okay, thank you, Ms. Cho. Thank you for the talk. Okay, our next speaker is Dr. Alex Figuera. Dr. Figuera is the Executive Secretary of the International Commission on Zoological Nomenclature, or ICZN for short. As the Secretary of ICZN, Dr. Figuera manages the daily affairs of the Commission, like handling correspondences and working with the Editor-in-Chief in publishing the Bulletin of Zoological Nomenclature. In his talk, Dr. Figuera will be sharing with us about what the ICZN is all about, its history, and its importance in ensuring that every animal has a stable, unique, and universally accepted scientific name. Let's welcome Dr. Figuera to the stage.
Hello? So this year is truly a year of anniversaries. We have SG 60, the 10th year anniversary of the Likongchen Natural History Museum, and it's also the 130th year anniversary of the International Commission on Zoological Nomenclature, or as it's better known, ICZN. And I've been the executive secretary for the last 2 years. However, zoological nomenclature as a discipline has been around for 267 years, beginning with the publication of Carl Linnaeus' 10th edition of his Systema Naturae, published in 1758, where he named over 4,000 animal species and nearly 6,000 species of plants. As Peter Ng mentioned earlier, he is widely regarded as the father of taxonomy, and his system of classification, where you place or group living organisms into hierarchical ranks, is still in use today. For those of you who don't know, nomenclature is just a system and rules for naming living organisms, and zoologists are most concerned with binomial nomenclature, where each species or type of animal is giving a unique combination of a genus name and a species name. And we're most familiar with Homo sapiens, which is the scientific name for humans. But how many species are there in the world? Current estimates place this number at 1.5 million described species, but every year there's about 20,000 species being described. But the true estimate of the number of species is upwards of 8.7 million. And since we're dealing with names, for these 1.5 million described species, there's about 385,000 genera names and between 3 and 4 million species names, which is more than double the number of described species. So you can see that there will be conflict and confusion regarding the scientific names of some species. So people will usually use common names to refer to species, but as you can see here, one species can have multiple common names or several common names. So the mountain lion or the cougar actually holds the Guinness World Record for the most number of common names at 48. And this is only English names. This doesn't include local names or names that are in dialect. Furthermore, common names or one common name can be used to describe two different types of species. So you could have cat-eyed snake, species that look probably fairly similar, but one is from South America, one is from Southeast Asia. But you can have things that are totally different, such as the fritillary, which could be a butterfly or a flower, or albatross, which could be a butterfly or a bird. So and most common names are ambiguous. So it's much better to use binomial nomenclature, where each species will have a unique combination of a genus and species name, and this will ensure that each species and their name is unique and universal. And this is what the ICZN focuses on. Their mission is to ensure that every animal has a stable and universally accepted universally accepted scientific name to make sure that they are unique and distinct. But the ICZN is not a legal institution. It's more of a widely accepted convention that scientists kind of buy into in order to make sure that the scientific names of animals can be easily regulated and communicated throughout the world. There's two roles for the ICZN. One is to maintain international standards of nomenclature, which they do by producing the International Code of Zoological Nomenclature. And the second is acting as advisor and arbiter to the zoological community by answering any questions that may come up either through emails or, mainly through emails, and by ruling on individual nomenclatural problems, which we'll get into. And as I said, it's the 130-year anniversary of the ICZN, which was founded in the Third International Congress of Zoology in Leiden in September 1895. It's important to highlight what the ICZN does not do, and we do not make taxonomic decisions. We do not give names to organisms or enforce the correct use of names, and we do not change names. But we often get letters from the general public asking us to change the common names of species, which we do not deal with. But some entertaining letters that we get is one like this, where someone wrote in and said that him and his girlfriend are unable to remember the name of the red panda, so they ask us to change it to cherry monkeys. Or someone else who says that complexus is a better descriptor than sapient for humans and ask us to change the name to Homo complexus. And one person who says that the name of the beaver should actually be the woodchucker, as they are the true titan of timber manipulation. And they even submitted a poem to go along with this. And they ask, what's a woodchuck chucking wood for? So the ICZN is currently made up of 27 commissioners distributed among 16 countries, and they largely do their work or perform their roles from their home institutions. Whereas the Secretariat, since its inception, has been based at the Natural History Museum of London all the way into 2016, where it moved here to the Lee Kong Chian Natural History Museum. And the reason for this is because it was largely being funded by a charity trust, and that trust ran out of money. So Prof. Peter Ng's work did not cease with building the museum, but also in saving the ICZN Secretariat. So he was able to get NUS to save the Secretariat from 2014 to 2016 by getting them to fund $100,000 for each year, and then afterwards setting up the ICZN Secretariat Endowment Fund, which follows a similar scheme as the museum, and the Lee Foundation is the largest donor with $1.35 million. So the code is online if you're interested in seeing it. It consists of 90 articles that have mandatory provisions, and these are the rules of nomenclature, and it just happens to be the 120-year anniversary of the Code of Zoological Nomenclature. So the code is meant to be a do-it-yourself manual. If you're able to resolve a nomenclatural issue, you could publish it in a regular research article, but if you cannot, and ICZN intervention is needed, then you'll submit a case. So really quickly, researchers will submit a case detailing the nomenclatural issue, and they'll list out a bunch of proposals and what they're asking the ICZN to rule on. And then this case will remain open for about 8 months to allow any comments to come in. Then those comments are subsequently published. Some of them will consist of counter proposals. And then after 4 months of the last comment being published, the case will then be presented to the commission for voting, and then the ruling is published as an opinion. The cases, comments, and opinions are published in the official journal of the ICZN, the Bulletin of Zoological Nomenclature. And now I just want to talk to you really quickly about an example case, which consists of the Archaeopteryx, which most of you should be familiar with. It is the earliest known dinosaur that's able to fly, and it's also regarded as the oldest known bird. Archaeopteryx actually stands for ancient feather, because the holotype, or the type specimen, was named after a feather. And in German, the name is also known as Urvogel, which stands for the first or primeval bird. the Archaeopteryx is important because it's considered to be a transitional fossil between non-avian dinosaurs and avian dinosaurs. And it was also, the description was also published 2 years after Darwin's Origin of Species, so it was widely regarded as proof of his theory of evolution. So the first case in 1970, the same time that the holotype, the feather that is shown here, was described, there was a more complete feathered skeleton that was found in the same year, but it was not seen by von Meyer when he described the species. So when that feather skeleton appeared, researchers started giving different names to this species, or I'm sorry, to that specimen, and then it was jeopardizing the name Archaeopteryx, the name that was associated with the first bird, and which is an icon of evolution. So in order to protect that name, these researchers wrote in, they submitted a case asking the commission to conserve the name Archaeopteryx and Lithographica. But 4 years prior to that, in 1857, von Meyer described another fossil which he believed to belong to a pterosaur, and he named that a pterodactyl. He named it Pterodactylus crassipus. And then that specimen was later found to actually belong to an Archaeopteryx. So in that case, the name crassipus will take precedence over the name Lithographica according to the code. So in order to not disrupt taxonomy and nomenclature, another case was submitted asking the commission to suppress the name crassipus and conserve the name Lithographica. And then finally, the feather is not representative of the species, so it cannot be used as comparative material to compare with other fossil specimens that may look alike or may look similar. So another case was presented in 2007 where they asked the commission to use their plenary power to set aside the holotype as a type specimen and designate the more complete feathered skeleton that was at BMNH as the neotype, which is shown here. If anyone is interested in Archaeopteryx, there was a new research paper published this year that shows a more complete skeleton, and it has a lot of the feather features and actually shows that Archaeopteryx was capable of flight. And although we'll never know what Archaeopteryx truly looks like, I urge you all to go and visit our gallery where we have a replica of the BMNH neotype. And this is going to be the cover of the next issue of the BZN, where in celebration of the 10th year anniversary of the museum, where we used the batik designed animals that you can see on our shirts and pins. Thank you.
Thank you, Dr. Figueira. We will now open the floor to any questions. Okay. We have a question at the third row. Oh, no question. Oh, no question. Okay, we have a hand there. Yes.
Okay, my name is Matthias Maywald, clinical microbiologist. I have two questions and I do apologize. One is, are there any nomenclature rules governing non-extant species, such as what you described, fossil species, that are different from extant species? And the second is there any repository for the names where people can orient themselves as to what the correct name is? For example, in bacteriology there is one, and for other species I'm aware of the National Library of Medicine taxonomy browser, but otherwise I'm not aware of a...
I'm sorry, I couldn't hear your first question.
First question was, are there any rules governing the naming of non-extant species, like fossil species, that are different from currently existing species? And the second one is there any repository, like website, etc., where nomenclature can be looked up and retrieved to be correct? For example, in bacteriology, my subject, there is one.
Yeah, so the rules of the code actually also applies to fossil animals. So as long as it's considered an animal, and actually if you look at the code, if anything is considered to be an animal by someone, then it will fall under the rules of biological nomenclature. So there was actually a paper, I think it was published in our last issue, where someone is talking about how do we go about naming extraterrestrial organisms if they do exist and if we do come across and find them. And to answer your second question, there is a repository. It's called the Official List and Index. So when a case is submitted and when a case is ruled upon, so first in the case, the researchers will lay out their nomenclatural issue, and if there's a name that they're trying to conserve, right, then they will ask that name to be placed on an official list. And if they're asking for another name to be not valid, then that name will be on the official index. So the ICZN collates that, and the last publication compiling the names was in 2012. So now it's part of my work to update that list, which I'm currently doing. And then we're hoping to get that up online, either on the ICZN website or on Zoobank, if you guys are familiar with that.
Our next speaker is Dr. Lim Swee Cheng. Dr. Lim is a marine biologist and curator of sponges at the museum. His research focuses on the taxonomy and phylogeny of marine sponges in Southeast Asia, as well as deep-sea sponges from the abyssal plains of the Pacific Ocean. Today, Dr. Lim will be introducing to us the fascinating and yet little-known history of sponges in Singapore. Let us welcome Dr. Lim to the stage.
Hello, I'm Swee Cheng. I'm the sponge curator of the museum. So today I'm going to tell you about the forgotten sponges of Singapore. So this photo was taken in 1910, a time where many Singaporeans knew about these sponges that we didn't know of, or rather that we didn't know of before I started studying sponges. And we're going to uncover the history of Singapore through the eyes of sponges in this talk. So this is a sponge garden in an intertidal reef flat in Singapore. So you can see it's beautiful, right? So there are over 10 species of sponges over there of different colors, size, and shapes. So sponges can be found everywhere in Singapore. In the intertidal reef flats, in the coral reefs, and in the deeper, on the seafloor in the deeper waters, and also in the canals and reservoirs. So I need to tell you about more about the fascinating sponges. So sponges are animals. They are the most primitive animals without organs or tissues. So they are very successful, over 9,000 species worldwide. They are sedentary, they cannot move or swim, and they filter feed. So if they cannot move or swim, so how do they protect themselves from predators? So they have a glass skeleton laced with unpleasant chemicals. So sponges are also novel chemicals and metabolite factories par excellence. So a lot of these products are used in antibacterial and antivirus drug. So each sponge species has a unique spicule and skeletal structure composition. So you can see on your left, those are spicules made of glass. And there are over 300 different kinds. And on the left, it's small examples of skeletal structures, which has infinite permutations. So a sponge species can have one spicule or more than 10 spicules and the combination to form so the combination that can be formed is countless. So is it countless? So let us go back in time. So in 1819, when the year that Sir Stamford Raffles founded modern Singapore, the Neptune cup sponge was discovered. And on the left, you can see the drawing of the sponge. It's 1 meter tall, you can see. So, and you can see so when I was studying sponges, the first time when I look at this, when I first saw this article, I thought there must be a mistake. How can this sponge exist in Singapore? Nobody has seen it in our waters. So probably the scale is wrong, or probably the locality was wrong. But it's true. So the museum, Natural History Museum, RMBR, before Lee Kong Chian Natural History Museum, we had specimens of these things, this sponge. So that made me believe it and strengthen my resolve to find this sponge. So, and you can see, during the first marine natural history expeditions in Singapore, the dugong was discovered together with the sponge. So did you realize if the first species that was discovered at a new place, so what does it tell you about the sponge? So it was abundant and common during that time. So Singapore during that time was a very different place with a different coastline. So there's a lot that we can gather from old literatures and specimens in the museum that were collected long ago. So this is something important that I want you to know. Oh, yep, sorry. So they treated that as entertainment that was truly marine. And this is John Crawford, the second British resident. So on 6 February 1822, he wrote those gigantic sponges which are peculiar to the coast of Singapore. So it's something that that's quite unique to Singapore. So you need to realize this, how iconic this sponge was to Singapore. But in 1908, in the guide to the zoological collections of Raffles Museum, written by the then director of the museum, Harnich, that was the last record of the sponge in 1908. So what happened? So the two most widely possible reasons are over-harvesting, because the sponge was really popular with the Europeans. A lot were brought back back to Europe to the museum or private collectors. So if you go to any museum in Europe or the major museum, there will be at least one sponge, Neptune cup sponge specimens there. London has about 10. Leiden has about 30. Yeah, so that was the popularity of this sponge. But I realized, okay, so this is to show you that the iconic status of the Neptune cup sponge. So this is a cigarette cut produced in 1928. In this series of cigarette cut titled Wonders of the Sea. Yeah, so that's when the whole world knew about this sponge, not just Singapore. And it was a quaint time when it's cool to take a bath and learn new knowledge about the marine animals. And another picture to show you how magnificent the sponge is or was. you can see a child sitting in this Neptune cup sponge. So this specimen is still around in the Natural History Museum in Leiden. So you can see it for yourself over there. So back to the reason why the sponge disappeared. So I realized after going through literature, so land reclamation begins very early in Singapore. So that's Telok Ayer Bay before Lau Pa Sat was built. So you can see. Yeah, so extensive land reclamation started in before 1900. And basically land reclamation is pouring sands into the sea. So we have a lot of silt in the water column. And remember I told you earlier that sponge was a filter feeding animal. So silt in the water will affect the sponges adversely. And this could be one of the reasons that that caused the disappearance of Neptune cup sponge. Yeah, so this is land reclamation over the years. So we don't see them for a long time. This was probably one of the reasons. You can see almost the entire southern coastline was reclaimed. So this began in the 1960s when Singapore gained independence. Yeah. Then there's someone that I need you to know. So this is Chuang Shou-Hua. So he was the head of the Department of Zoology in the University of Singapore in 1970. So this is, so you can look at the letter there. So he made a deliberate search for the Neptune cup sponge for over 30 years, but he didn't find the sponge. So these letters were discovered when I made a trip to Yale Peabody Museum because Prof. Chuang collected over 100 sponge specimens from Singapore in the 1950s, before land reclamation or before Jurong Islands were reclaimed. So those are important collections that tells us a lot about our marine environment. So after preliminary examination, I realized some of the sponge are not present anymore. Yeah. So Prof. Chung is still, so he was born in 1990. He's still alive. He's 106 years, and he's still very active. In fact, he published a seminal book on brachiopods in 2016 at the age of 97. Yeah. So he made a huge contribution to the marine biology studies in Singapore. So 2008 was when I started my studies, started to study sponges. And so on that page 66, that was when I did my literature research, found out about the sponge descriptions and these photographs. So I made a call to the biologists in Singapore to keep a lookout for this sponge. Yeah. And this leads to the second forgotten sponges that I want to tell you about. It's a buff sponge. So I found this sponge. I didn't know what is it. So finding a sponge it's like finding a present. You need to open it or examine it before you know what it is. Because sponge are variable in form, colors, and shape. So so you need to examine it, to look at the spicules, the skeleton, before you know what is it. So that's a buff sponge. So after examining it, I realized it doesn't have spicules. And yeah. So bath sponge. So the first bath sponge was recorded by a German student in 1905. And you can see by a report from Willie Mott, a British officer, fishery officer. The bath sponge was packed in cellophane for sales in Chinese shops. But we don't have this culture anymore. And in fact, they were serious about sponge industry. So we had that much bath sponge in the past. Yeah. And to 2011, the Neptune cloud sponge was rediscovered. That was me, looking quite calm, but my heart was pounding very fast. There's a baby sponge there. You can see it's like a stool, and the cup was not formed yet. Yeah. So, yeah, thank you for your attention.
Thank you, Dr. Lim. In the interest of time, we will skip the Q&A, and then if you have any questions for Dr. Lim, you can ask him during the tea break. Okay. Okay, for our next speaker, we have Mr. Foo Maosheng. Mr. Foo is the curator of insects at the museum. He also manages the museum's cryogenic repository. In his talk, Mr. Foo will be sharing about the discovery of Nocticola pheromosa, a new cockroach species in Singapore that was described in 2023. He will also highlight Singapore's rich insect diversity and show how incorporating pop culture, like Pokemon, can foster public awareness of Singapore's biodiversity. Let's welcome Mr. Foo to the stage.
Hello, all right. Okay, just before that, I think I will just do a safety announcement, right? If anybody here who's afraid of cockroaches, okay, so there's quite a few of you. Why? It's normal, but let you know, there will be a number of cockroaches that will be appearing on screen. So if you are still squeamish about cockroaches, maybe just cover your eyes, right, and just listen to this as a podcast. So, if you are ready, I'll start. So yes, today we'll be talking about cockroaches. I'm sure that most, if not all of you, will never dream of a day that you'll be entering an auditorium and listening about cockroaches. Because the moment you hear the word cockroach, there are two pesky cockroaches that will come to mind. These ones, do you recognize who they are? That is your American cockroach and your German cockroach. Two pesky cockroaches on a global scale that people will run away or even take a flamethrower and try to burn them all. Right, and in Singapore, cockroaches are actually one of the top feared species, more than death, than even being lonely, or even more than ghosts. But today, we are not talking about pesky cockroaches. We'll be talking about a cockroach that has eluded our eyes for many, many years. Right, this cockroach is known as your delicate cockroach, or previously we call it as the cave cockroach, something I will explain later. But to the public, they know it as the Pokemon cockroach. So yes, as you have known from the earlier speakers, that Singapore is actually teeming with lots of life, despite our urbanized landscape, our small forests. Who knows that it actually houses a particular small critter that is actually going to a global phenomenon, right? So who knows about that? But the question here is, how did it actually happen? So let's have a quick recap. So it was actually part of the Bukit Timah insect survey, where we actually went to see what kind of insects are actually living in Bukit Timah. And on screen, you can get to see some of the colorful, charismatic ones to some of the strange and tinier ones. Now, sorting through all the insects, I would focus more on cockroaches. And that is basically this one. Now, when I first examined it, I was kind of curious because it neither looks like a cockroach, but at the same time, it looks like a cockroach. If you can see on screen, the cockroach is actually covered with hairs, very unlike with the other cockroaches that you have already known. So I take up and look at the identity of it, and I find out it belongs to a taxonomic family that has not been documented in Singapore before. I'm like, hey, this is actually very interesting. So what we do next, right? And just to show you an idea of what we know, we previously called it as a cave cockroach. This is actually how it looks like. pale body, long antenna, and the one unique feature about for those that are living in dark caves is the almost negligible eyes that you see on screen. So, all in all, we decided to do some sequencing, right, put it up online, and then maybe wait for any particular expert around. But not very, it was quite fast. And then we caught the attention of a Filipino entomologist who actually wanted to examine the cockroach. So I thought, hey, why not pass it to him? Right? He has the better expertise than me. He wanted to do a genitalia dissection because on the surface, the cockroach cannot tell you what species it is. So, all right, send it to him, and he did the genitalia dissection. Now, take note, the cockroach is only the size of a rice grain. And having to do genitalia dissection is on a whole different level. So, with the genitalia dissection, we know that it's actually a new species, completely different from the Borneo species that we have in Southeast Asia. So, when we know that, hey, this is a new species, of course, we started thinking, what should we actually name it after? So, our first choice was actually the first director of the Raffles Museum, right, Dr. Carl Richard Harnisch, because he too is an entomologist with a particular interest in cockroaches. So we wanted to actually name it after him because to honor for his works and his contribution. So okay, that's our first choice. But then as we talk more, we look at our manuscript, we realize we are both avid fans of Pokemon. We are literally the bug catchers in the Pokemon world. At the same time, we also do acknowledge we are both scientists who actually look into groups that is highly stigmatized, that incites fear, that incites hatred, and also has a significant impact on the public economy and health. And as we talk more, we also realize we have the same goal, right? We have studied this particular group for quite a number of times and number of years. And we know that this particular group of insects, they are not as bad as what they seem. So we also do want to shift the perception, not change, because changing the perception itself will have to undo many, many generations of trauma. So in short, we decided to actually name it after Pheromosa. Now, who is Pheromosa? Pheromosa is this particular character from the Pokemon world. Now, interestingly, Pheromosa the design was inspired by the freshly molted American cockroach. So you can see the uncanny resemblance. So in short, we have Pokemon that is actually inspired by a real-life cockroach, and now we have a cockroach named after a Pokemon. So some of the avid fans rename it as a full circle moment. But is this the only one that has been named after, say, a pop culture? No. There are actually more, right? It's a kind of, you say, as a potential trend in taxonomy. So one other example is a particular famous person who was in Singapore this year. Anyone can make a guess? Right, Lady Gaga. Lady Gaga has also have a tree hopper named after her. And how about last year? Who's the famous person? Correct. There is a millipede named after Taylor Swift too. And then if you also know, we have also a species that's named after Shakira. Now, this Shakira wasp does not do the belly dancing. It actually causes the caterpillar to contort in a way that it looks as if it's doing belly dancing. But these are not the only ones. There's more. Beyonce fly. Right? You see the big golden bud. But do you know, the horsefly is actually a bloodsucker. Okay, I'll stop there. Now, there are also other insects that are also named after Pokemon. For example, known as your Charizard bee, as well as the one before Pheromosa was the three legendary rare beetles in Australia, right? Named after the Pokemon of Articuno, Moltres, and Zapdos. And also, not forgetting our marine folks, this year we do have the giant arthropod named after Darth Vader. Now, if you are not sure who Darth Vader is, this is Darth Vader. Okay, so you can see the resemblance to it also, right? So we do have species named after different celebrities and pop culture. I also did a survey, which some of you may actually have participated in, right? Whether does this actually causes a change of impression, especially with regards to cockroach. And I could say positively, yes, there is a change, but more can be done so. Now, why do we actually name species after some of the famous celebrities in the world? First one is looking at the shifting of perceptions. We do know charismatic species are actually, taken the limelight for many, many years. What about the others that are non-charismatic? They too also have a value in our ecosystem. And also public engagement also, right? All of you here are actually, I would say, biodiversity or nature inclined. How can we actually reach out to the greater masses who actually maybe, say, stuck to their computers, stuck to accounting, stuck to finance, stuck to engineering? How can we reach out to them? So this is actually why we actually named them after pop culture, right? And following up on my survey, I also asked those who take the survey is whether does this actually engage them even more. And results shows that it actually does. Oh yeah, so if you actually look up online, there are actually many other species named after different celebrities, including Barack Obama, Andrew Garfield, and Angelina Jolie. So all in all, does it actually matters, right? Because one thing you can know is that it actually is beyond the hype. When you actually name after a species, you can actually go about protecting, as what Prof. Peter has mentioned, right? You cannot protect something that you do not know that exists in the first place. And also because of shifting perception, this particular group of cockroaches, they do have a value in our ecosystem also, right? As you know, cockroaches, you have to think, oh yeah, they're pesky, they bring bacterial infections, they cause diseases. But cockroaches, they also do, say, have a role in our ecosystem, recycling nutrients, and even doing pollination. And last but not least, is actually having to draw new attention, right? Taxonomy-wise, right, you guys might be a bit familiar, but it's also, in a way, could be a dying science in some sense. So how can we actually continue, actually inspire the next generation to actually go about continuing our work, right? And basically try to get them to relate to what we are doing. So if Singapore, in such a small country, can actually house an interesting cockroach, what other marvelous creatures can we actually explore? I'll leave up to the next generation. So anyway, to end off my talk, right, basically if you want to look at, say, naming them after pop culture, it's basically changing the perception, gaining new attention also, right? So once you gain a new attention, people can actually have a better relation to what they are looking at. Right? For example, you're looking at the cockroach, people will get to relate to the Pokemon game they are playing. And from there, we can actually foster better understanding and respect for our biodiversity. So, and with that, right, I'll keep it short because I know it's cockroach, right? Don't want to hear too much. I actually am appreciative that you have joined me on this particular journey here. Right, thank you.
Thank you, Mr. Fu. We will now open the floor to questions. Questions, anyone? Okay, Mr. Fu, I have a question. So out of curiosity, are there any challenges in naming species after like celebrities, like copyright issues?
Oh yes, I do get that question a lot, yes. There can be, say, challenges to it, definitely, right? But sometimes they don't want to be associated with that particular insect or something, right? For example, like say you're looking at the cockroach, right? If I were to say name it after some people, they might think that, oh, they have the reputation that they are dirty or they are pesky itself, right? But yes, there will be some challenges. So sometimes you may actually do have to seek, say, the permission or say, to let them know that we actually want to name it after them.
Oh.
Yeah.
So that means you guys actually reached out to Pokemon on...
In that case, actually, not exactly. Not exactly. Yeah.
Thank you. Questions, anyone? Okay, I have one more question. So, what inspired you to be an entomologist?
I would say it actually started when I was in kindergarten school. Yeah, in kindergarten school. For those who have known, right, my first pet is actually a cockroach. Yeah, so it's from there itself I get to find out, insects are actually more than just creepy crawlies. They are actually quite interesting, right? They have lived very different kind of lives, completely different from us humans. Yeah, and that's actually what fascinates me. So, and regards to very similar to what I do, right? I'm always very curious about what is actually out there. What new species can we find, especially for a country that's so small in Singapore? There are still more to be discovered. All right, thank you.
Thank you, Mr. Fu. Our next speaker for the afternoon is Dr. Ang Yuchen. Dr. Ang is the Senior Curator of Insects at the museum. His research focuses on dark taxa, which refers to groups of organisms that are species-rich yet largely undescribed and understudied. In his talk, Dr. Ang will highlight how DNA sequencing and artificial intelligence can help us discover species at a rapid and rigorous rate. Let us welcome Dr. Ang to the stage.
Hello. So today I'm actually going to be talking about a bit more technical stuff. So this is what I call dark taxa, a lot of insects, and then how do we try to illuminate them, okay? So, I'm like, actually this, I've been introduced, I'm a curator of entomology, and I actually like to work on flies. So there's one cockroach, there's some who worked on bugs, and then I'm going to talk a lot about flies. Okay, so what is dark taxonomy? It's not like dark arts or anything, okay? It's actually, I think, because I think we have a lot of talks that talked about what taxonomy is, so I'm just going to breeze through. Okay, it's really about the naming, describing, and classifying a species. It helps us answer, what is it? Is it new? Is it different? Like that new cockroach, right? Okay, so as a taxonomist, you go out there, you catch stuff, you try to analyze them, you publish papers so they become actual species. Okay, where's the dark part coming from? Before that, I'd like to play a game. Okay, ding, 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 ding. Roughly how many species of terrestrial plants and animals can be found? Oh, shucks. You saw it, 50,000. Thereabouts, because there was actually a question that said, how many species do you think are described from Singapore? Well, that's actually it. So let's go a bit more into it. Okay, Singapore's so tiny, how can we have 50,000 terrestrial, not including the marine organisms? So let's see, our known biodiversity, about 4,000 plants, and about almost 800, give it 800 vertebrates, okay? Charismatic insects like the butterflies, the dragonflies, damselflies, about 400 plus. And what about the other 44.5K? These are all the smaller groups. Actually, we already have talks on the bugs, we have some beetles, okay, and then the ants. Wendy was giving talk on the ants, so I'm going to talk about these buggers here. They are the biggest group here. Dark taxa, what are they? Very specious, okay, in what we call taxa. Taxa means it's a technical term for a group of species, okay? So they have, in a dark taxon, there are very high number of species, very abundant as well. So each species may have hundreds, thousands, okay? And they are very poorly studied, okay? So as an example, these are all these are new species which are pending description, okay? They're tiny, abundant, very diverse, and usually tend to be tiny, uncharismatic, little spots, okay? Very hard to differentiate. Maosheng mentioned how he had to do genitalia dissection. Kind of has to be done here as well, and my guys are like half a grain of sand, okay? Some of them are so tiny, okay? Very similar to each other. So, because of all these issues, right, we do realize that after 273 years, this is the number of valid extant species, basically the number of described species since all the way from the start of Linnaeus to year 2025, okay? Not more than 500 a year, okay? So, why are there so many undescribed species? Let's take an example. Let's look at these group of flies. They're called mycetophilids. They're pretty slender flies. They don't really, when you think of flies, you think of those big, fat, buzzy fellas, right? But these are mycetophilids, which means fungus gnats. They are very, very tied to the ecology of our fungal ecosystem here, okay? They need fungus to breed, very sensitive to the environment. So basically, they can be good indicators for our fungal ecosystems. So, we did a series of nationwide surveys since 2005, okay? These are what we call Malaise traps. The flies hit this little like a tent. They will tend to go upwards, and then in there is a bottle of alcohol. They smell the alcohol, they get a bit drunk, and poof, they all go in. So what happens? We put all these traps in mangroves, swamp forests, Bukit Timah, okay, secondary rainforests, okay, university campus, we're starting to get desperate, right? Some people's homes as well, okay? All these traps, what happens? After a week, this is usually what you see. How are you going to deal with that? Okay, so in all, we had about 6,500 specimens, okay? What next? How do you do? Okay, so the traditional way is to sort, okay? You get a whole bunch of interns, you get students and interns, you get one expert there to like, yeah, no, you got that wrong, no, that's not a fly, that's a beetle, things like that, okay? Then it gets pre-sorted in all these little containers, okay? And then we send it to expert, usually some old, tired Angmo in the States or something like that, all right? I'm not trying to be ageist here, but the fact is most of the taxonomists are old and aging. There's no one to replace. Okay? Now, the taxonomist himself, he can't remember all these species, so he has to use something like a key. So a key is something like this. You read the first, what we call a couplet here, and then, yeah, no, this one, let me see, is it this? No, blah, 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 blah. It takes about 5-10 minutes. Okay, I think it may be this one. Then he has to go to the species description and check. Oh, is it this? Do I confirm if this? Blah, blah, blah. Oh, I think it is. Okay, is that species? Okay, ID confirmed. A lot more to go. So that doesn't work, right? It's tedious and then subjective accuracy. If the old guy is like, he drinks a bit too much or he's a bit too sleepy, he misses something, you're in trouble. So, this is what we call conventional taxonomy. It's actually optimized for I call light taxa, okay? Moderate number of specimens, lesser species, and then very well-known taxonomy already, okay? The knowledge is well done, okay? Usually what I call morphology-based, that means looking under microscope. And perhaps this is good career advice for people to start. So why did I go into something like that's dark taxa? It's a bit insane, okay? Dark taxonomy. So in a way, I like to present a set of scalable protocols to study the dark taxa. Oh, there's a screen, yeah. So what I do like to use is what I call a reverse workflow, okay? And then first we use DNA, we sequence everything, okay? And then we use DNA to help us pre-sort. Then after the pre-sorting, it's easier for us to use morphology to check all the different clusters, okay? So the goal is then to speed up the discovery without sacrificing the quality. Now, let's go through it. How do we do that? For example, why is it not working? Okay, so these flies are pulled out, okay, they're put into this DNA extracting well. So each specimen goes into one of these little wells, and then I'm going to skip the technicalities, it's sent into a DNA extraction process. We do the sequencing. So in the end, what happens is we have vouchered specimens, okay, and then the DNA is matched to each of the specimens. So these are all actual DNA sequences, okay? So I take an example out here. So specimen A to H, these are all the different sequences. And actually you do see like, look, there are some differences here and there, right? So we try to match them according to the sequences, okay? And these are what we call clusters of species. Next, I use this information to actually group them into what we call putative. That means we think they are the species. So these little baggies there, I look like a drug dealer here, but each of them are like a few vials of specimens. Okay, we do imaging. Okay, so I'm going to skip this because SIGNIFY has actually gone through all this, how we actually do our imaging. We image in certain layers, we combine them into a properly focused and high resolution image. Then we put it up onto the Biodiversity of Singapore, okay? So every time I have new species or putative species, I like to image them, I put it up on the BOS, and then people get to look at it, okay? So these are all the mycetophilids, the fungus nets have been imaged. And I bring my colleagues over, actually a lot of old Western dudes, okay? And then we do the species description, okay? So these are all the plates of images that we've been done, 115 new species, okay? It's still in publication process, and we have a problem. Maosheng has one species to name, we have 115. What are we going to do? These are all the names. So we had to get creative. We tried to have variations of names of Singapore, and my colleagues suggested, actually, good idea, why don't we go to the Singapore Women's Hall of Fame, and then we tried to, utilize that. But I'm going to run out of names thereafter, because we still have a lot, at least 300 more fly species to name. So help. Okay, so that is the molecular sequencing part. What about robotics and AI? Actually, the museum is embarking on a project to try to develop a framework where we can utilize AI as a kind of like co-pilot, not to take over a taxonomist's job because it's just too complex, not at this stage, but at least to help us with the more mundane tasks. What can AI and robotics do? Okay, first of all, there's this, actually this is already published, there's a machine called the diversity scanner. So it's actually a machine that is able to take a look. You put all the specimens in here, okay, it has a little camera, and then it will detect all the individual insects. It will take a photo, and then it will put them, suck them up, and put them into a sequencing well. So instead of me doing all the pulling out of the specimens and putting them into a sequencing well, it does it automatically and generates a photo. Okay, and with the photo, we throw it into some AI networks like CNNs. Okay, what happens? It actually is able to, varying degrees of success, because it's still starting stages, to try to identify them to at least the families. Okay, it's actually pretty decent now. Okay, so this is the first, some terms you've heard of, you may have heard of this AI, it's called convolutional neural network. These are AI models that are really good at categorizing objects, images, But they cannot explain or translate the results. They can categorize for you, they can't explain why, So here comes the next step, NLP, natural language processing. It makes sense of highly variable taxonomic texts. I'm taking a step back here because we have an existing huge amount of knowledge that is not being tapped by us people and by even machines, All these species descriptions, they can be in different languages, And even, this is a scan of Wallace's, his notebooks, and has actually good information, habitat information, for example. All this, NLP, as AI, natural language processing, can help to extract all these kind of what we call key semantic data and compile them into a readable format so that machines, AI models can begin to utilize them. And also, of course, it can translate, right? So now every time you Google Translate, that's pretty much a lot of AI. So after that, I combine both the image and the NLP portions into what we call a multimodal large language model, It's able to bridge, then combine the images and text to become truly a bit more smart than the lower average intern, which will help, So it then starts to serve as interactive teaching tools, making the expert logic visible. So instead of having like a teacher to teach you, you can capitalize a bit on a robotic brain to help us to a certain level. Again, I emphasize we're not replacing taxonomy because you always must have humans in the loop. Otherwise, trouble happens. So the project we have is we want to try to design a co-pilot, the multimodal large language model to be lightweight and locally adaptable to help ASEAN scientists in the region. So instead of getting the old Angmo, we can duplicate this via the AI platforms. And that's it. Thank you.
Thank you, Dr. Ang, for the talk. In the interest of time, we will be skipping the Q&A. If you have any questions for Dr. Ang, you can look for him during the tea break session. Our last speaker for today is Dr. Tan Yen Yi. Dr. Tan is the curator of birds at the museum. Each year, millions of birds around the world die from collisions with man-made structures like tall buildings. Many of these deaths go unreported, yet each hold a piece of a puzzle that scientists are still trying to unravel. In her talk, Dr. Tan will introduce to us the Dead Birds Hotline, a citizen science initiative where dead birds are reported by members of public and collected and preserved to serve as reference material for research in future generations. Let's welcome Dr. Tan to the stage.
Good afternoon, everybody. As Norman has kindly introduced, I'm the curator of birds at the Lee Kong Chian Natural History Museum, and today I'm talking. My talk is titled "Dead Birds, Living Data" because it's a catchy title, and also I would like today's talk to impress upon the audience how dead animals actually contribute to a lot of data that we use in our daily lives. So, without further ado. So when people think about ornithology, ornithology is a big word. So sometimes when little kids come up to me and ask me what I do, I don't tell them that I'm an ornithologist. I say I'm a birdologist because it's much easier for people to understand. But when people think about ornithology, they tend to think about this, right? You work with cute, fluffy birds. And unfortunately, that's not always the case because as many of us here, when we study zoology, it's not just about the live animals that we study. Yeah, and this is me enjoying that part of ornithology. But what people don't think about is that we work with a lot of dead material, a lot of dead animals, and this is not always pleasant, and it is actually a lot of the dirty work that is necessary, essential to today's science, but nobody really wants to do it, and it's not something to be loud and proud about. But I do think that we should be loud and proud about it because it's the bread and butter of a natural history institution. So my work has been covered by various media sources, and it's something that helps to promote what our museum does, not just what I do, but also what our museum does in general. So I hope you guys can go look up these articles and have a read too. So many people ask me, sometimes when members of the public, they text the hotline. The hotline is me, by the way, so if you drop a message to the hotline, it's invariably me. It's just me. No one else is running it. So I have people asking me, the bird died already, got used, meh? I'm here to tell you, yes, got used. So out of the many specimens that we have reposited at the museum, as the curator of birds, I'm just in charge of that tiny little section over there. This figure is from 2010, so it has since increased a little bit. We now have about 34,000 specimens, bird specimens in our museum, representing about 1,000 species and counting because natural history collections never really stop. Why are natural history collections important? I'm sure many of you in the audience here can already appreciate why the museum is important because we are a center for biodiversity research. We hold the resources and expertise necessary to further this scientific endeavor, right? So what we do, and what I do also, primarily as a curator, I want to maintain a collection that is that can serve as valuable reference material for now, later, and hopefully forever. It serves as a timeless repository for species that maybe are no longer with us on this in this mortal realm, right? So here are examples of two extinct species that I photographed when I had the opportunity to visit the Cornell Lab of Ornithology 2 years ago. And some of you may be familiar with the one on the left. So if you are a BAS student like me, you will know this species because this is a species that has been talked about as a poster child for extinction. The passenger pigeon used to be so numerous, innumerable in count such that when they flew in flocks, they could cover the sky and block out the sun, and yet they were hunted to extinction. And it only lives on in natural history repositories such as this one. And then if you're a birder, you'll be familiar also with the one on the right, which is the ivory-billed woodpecker, the holy grail of North American birding. It's said to be extinct already, but scientists are still quite reluctant to label it as such because you do have to wait for a few, about I think 50 years as a general rule of thumb of non-sightings, non-confirmed sightings before you can even consider something extinct. If you have just one uncertain sighting, it's still we still err on the side of caution and we don't declare something extinct. Well, it depends on the gender as well, but not to go too much into that. As a museum in Singapore, we also have valuable records of species that used to breed in Singapore but no longer do. These are what we call extirpated species. So this is one example. This is a very brightly colored bird. If you're a birder, you'll be familiar with this bird too. It's the green broadbill, very cute, very fluffy, has a very fluffy face. And we do have a few specimens collected from Singapore. And the last sighting of this bird was August 2021 on Pulau Ubin. And this bird here was collected, I'm not sure if you guys can see the locality, but it says Woodlands in Singapore. Okay, and then we also have the black and red broadbill. This is a slightly common bird, common rare visitor, a bit of an oxymoron there. But it was last sighted just last month on Pulau Ubin. And this specimen was collected on Pulau Ubin. Okay? And we also have many, many more representatives of has-been breeders breeding birds in Singapore, such as the rufous-collared kingfisher. I think this specimen is now on display in the Science Centre exhibit. And the red-naped trogon, very colorful bird. We used to have 3 trogon species that breed in Singapore, but no longer do. And then the whiskered tree swift and the rufous-winged philentoma. So we have records. These are irreplaceable biorepositories that kind of serve as irrevocable proof that these birds used to be here. Yeah, and our museum, the last I looked at our catalog, our museum holds represents about half of the extirpated species. So actually there are some specimens that we don't have of, and we cannot go back in time, right? And this is one of the examples that I like to tell people, like why it's important to collect. Because if you when you collect a specimen, it's not for today, it's not for tomorrow, it's for way further in the future. Just like what Prof Ng has said, it's a slow burn. And we may not even be here when the burning is happening. Yeah, and besides just physical repositories as physical specimens, we also, nowadays with the advancements in genetic technologies, genetic sequencing technology, we also have the possibility of conducting museomics. That is the extraction of genetic material, usable genetic material from museum collections that may be centuries old. So one example that I like to point out is the chickens project. I'm not sure if some of you may have heard about this, the study of red jungle fowl in Singapore. So this study conducted by my colleague Melody Wu, who is now pursuing a PhD elsewhere, she used some material from the Likhongqian Natural History Museum. She extracted DNA from our very own red jungle fowl, representing populations that used to be on the island maybe 80 years ago, and compared them with modern day samples of the chickens that you see running around Bishan, Ang Mo Kio, Tanjong Pagar, Outram, all these places. And what did she find? She found that the ancient red jungle fowl was actually very clean. The genetic profile was very clean. But modern day red jungle fowl always invariably had a fraction of domestic input from the domestic counterparts. This means all the mutts of the chicken world who are running amok have been interbreeding and contributing genetic material to our original red jungle fowl and are already eroding away at the genetic integrity, and also the appearance changes with this genetic integrity. That was something that she proved in her paper. And now there is actually a guideline on how to diagnose from the appearance of a chicken whether or not it's how pure it is with respect to the ancient, our original, the OG red jungle fowl. So I'll give a quick crash course here. So if you guys see a chicken, a red jungle fowl, okay, and it has white leopards, that means the cheek patches are white. It has gray feet, gray legs, no other colors, gray legs, black primaries, and olive green body, and a very nice long green tail. That is a pure red jungle fowl. And you cannot discount any single diagnosis criteria. Every single one of these landmarks have to be there. If not, it is a bit not pure. Okay, not to say that we have to conduct a genocide of all the red jungle fowl in Singapore. That's not what we're saying. But we are putting forth the fact that introgression between the domestic and the original red jungle fowl is happening. And if we want to stop it, then we have to think about ways of how to do it. Okay, so it's an often overlooked resource because museomics is still an evolving field. It's still quite new. But as we go, as technology advances, we can expect for museomics to play a much bigger role in conservation and genetics as we go. And then specimens, physical specimens themselves can also give you data that you can, that's very hard to obtain in the field. So the photo is a bit small here, but I would like the members in the audience to take a look at this specimen in the jar and tell me what's unusual about this specimen. So as a hint, birds only have 4 toes. I'll give you 5 seconds. This bird has 5 toes, which is a bit scary. And also the toes are arranged a bit like a human hand, and very unlike a normal blue-crowned herring parrot, which this specimen is. A blue-crowned herring parrot usually has two toes facing the front and 2 toes facing the back, but this one has 5 toes facing the front, and that was really creepy when we discovered it. This is a bird that was salvaged through the hotline as well, and with this specimen, it would be possible to conduct many more analysis in many more dimensions, such as we can try our best to chase the genes responsible for this mutation, and also to study the morphology of the specimen, because we have this physical specimen here. So this is to impress upon you that specimens are important, and they're useful, and they're interesting. And then one story I like to tell using our specimens here today is these common buzzard specimens. So common buzzards are considered a vagrant to, are not very common and are considered a vagrant to Singapore. These 2 specimens actually were collected way early, like in the early 19th century. And then a few weeks ago, we got an email from NHM UK, the London Museum, and they say, hey, guys, we found these 2 birds in our collections, and they have labels that say that they're on loan from you. Do you want them back? And then we said, yeah, sure, why not, right? And they sent it back with photos, of course. And these are the photos that they took. And you will see there that on this green tag here, it says on loan from D. Wells, Raffles Museum, Singapore. And those of you who know ornithology, D. Wells is a big name in regional ornithology. That's David Wells. Yeah, and he's responsible for this book, Medway and Wells, 1976, talking about the 5th installment to the birds of the Malay Peninsula. Okay, and these birds are significant because back then, not many of these specimens existed in the region. And when I emailed David, because we have his contact, and I heard from some people that he's maybe still around, so I dropped in an email and said, hey, David, we found these 2 birds we just got these birds returned from the UK, and I'm not sure if you remember, but they're back, yay. And then I didn't expect a reply because I know that David is very old. So there's a chance that he may never reply, right? But he did. The very next day he replied. And he said, oh, Yanni, that's great. And he told me, he recounted the whole story as to why they were sent. And he said, oh, we were writing this book. And then back then, no birder in his generation had ever seen these birds alive. And these were the only specimens that existed in the region. And so back then, he consulted Mrs. Yang, who was the custodian of the ZRC, the collections. And they decided that the best plan was to send it to the UK, so that they can conduct comparative material, where there's comparative material to study them. And then, and so from there, we started a little correspondence, and I thought that would be a cool story to share, because this is literally a piece of history that we have, right? So specimens are way more than just records of animals. Okay, and then I have to breeze through. So sometimes you find interesting things on labels. So if in Mandarin, the words in the red square say long mouth, suck suck, which means that whoever collected this bird was collected in China, and whoever collected this must have thought that these birds use beaks as a suction and use suction as a strategy to feed. But now of course we know much better about these birds, and we know that they don't do that. But it's just interesting to see how humans, where birds live, and how human society interacted with them. Okay, so of course, vertebrate collections are on the decline because of the ethical considerations that you have in contemporary times. So we actually do have some really old specimens in our collection. So when I looked up what's the oldest specimen we have, because our curiosity is this bird that's said to be collected in 1808, then some of you will say, oh, but the O may be a 9 or something like that. Maybe it's a typo or transcription error. Okay, fine, I'll find the second oldest one for you. So 1819, which is a slightly more reasonable time frame, I think, and a series of oriental predincles from Malaysia. How about the oldest ones from Singapore? We have 2 Dorian starlings collected in 1879, so these are the earliest birds collected in Singapore, followed by a booted eagle and an eastern barn owl. Okay, so and the majority of our collections are from pre-World War II, and which is why, and we have a big temporal gap between then and now, which is why we go with salvage. Because nowadays we don't go out and we don't collect actively anymore. And birds are dying by the thousands. We don't have to kill them anymore. The buildings do it for you. So what do? We just go and we pick up their bodies as they fall. Okay, not as they fall, but as they are reported. So every report comes into us. We assess whether or not it's worth attending to, and then we collect it. Okay. So shameless publicity, please save our number. And if every report is important to us, even if we don't collect the bird, it contributes to a digital repository of data. And if we do collect the bird, then it goes to our museum and becomes an irreplaceable part of history. Yeah, and from this hotline, we've gotten first specimens for Singapore, such as this Eurasian sparrowhawk and the short-tailed shearwater. And then just a few days ago, we got Singapore's first green imperial pigeon. Yay! This pigeon is not uncommon. It just doesn't die very much. So we can't go out and catch it and sacrifice it without the proper NParks permits. So when it's reported, we were like, oh gosh, we have to get it. So we sent someone to get it, and we did secure it. So happy to say that. And we just got a publication out in Birding Asia about Singapore's first record of this subspecies of hooded pitta. And if you're in the birding scene, you know that right now there's one hanging around in Simei. So if you live around there, you can go and see it, because this subspecies has never been recorded in Singapore until last year. And then now it's getting more and more common. So here's the time to document everything. And then we also, through the hotline, we also get a lot of records that are difficult to obtain otherwise. And every record that I get that's rare enough, I will submit it to our colleagues at the Singapore Bird Database. So a very good public repository. So if you guys are interested in birds, I highly encourage that you look up the Singapore Birds website. Okay, and of course, fortune favors the bold. Fortune also favors the boldly colored. So all the birds that are really colorful, they tend to be reported a lot as compared to something like that. And that's not to say that the Javan minor or things that are drab is unimportant. They are all important. So whenever you see a dead bird, just report it to us. Don't even think, okay? And of course, these serve as currency for international network and collaborations. I exchange specimens with other institutions to build goodwill, and also as an insurance for our own specimens. In case something does happen to our collections, then at least we can rest assured that there's other material that's reposited elsewhere with our colleagues that we trust. Okay, I've built connections with the Berg Museum and Cornell Lab of Ornithology. We invited them over to look at our birds and prepare some, bring some home, and exchange some with us. And also with Yale University, and with all the foreigners who come to Singapore, you always bring them to scale our highest peak, that is Bukit Timah. So they were very excited. They thought they were going up a real mountain, but I told them, no, please, it's really only 160 meters high. Don't expect too much. And they still had a good time. Okay, and then we also use the material to build capacity. All the dead birds, but no one to process them. Now we're trying to teach and teach this skill set and impart it to my colleagues, and also hopefully the next generation, so that one day if I'm gone, this continues. Yeah. And every bird tells a story, but only if we let it, if we help it tell its story. And I hope that you guys can all save my number again. And thank you very much.
Thank you, Dr. Tan, for the talk. In the interest of time, we'll be skipping the Q&A. If you have any questions for Dr. Tan, you can look for her during the tea break session. Okay, we will now be having the 15-minute tea break session. But before that, could I have everyone remain in your seats for a quick group photo? So for those at the sides, could I kindly ask you to shift towards the middle so that all of us can be seen? Okay.
Thanks for staying till the end of the day for this last session. This will be an interactive dialogue between the selected members of our research, biodiversity histories, and facilities team, including our senior management here. I am Marcus Chua, the curator of mammals at the museum, and I will be moderating this discussion with the aim of peeling back the curtain to show what's behind the scenes at the Natural History Museum of what our researchers do, and I'll try to make them reveal secrets on their job, and also we hope to achieve, in the grand scheme of things, of what we do as a Natural History Museum. So the members of the panel here are, first from left to right, Deputy Head of the Museum, Associate Professor Hwang Tan Wei, Assistant Head Dr. Tan Swee Hee, as well as Biodiversity Histories Lead, Mr. Martin Loh. These three distinguished gentlemen here were not part of today's speakers because they are reserved for this session. So the audience here might not be so familiar with what they do, so I'll let them introduce themselves in 2 minutes, please.
Good afternoon, everyone. My name is Dan Wei. I'm the Deputy Head So basically, I do whatever Darren tells me to do. Essentially, I'm also in charge of or rather oversee the research collections and curriculum section of the museum. That accounts for about half of the staff members. So we have about 50 staff members, about 25 are basically doing research, handling the collections and teaching generally at the university level. So that's basically my main job. My part-time job is to work on corals. That's actually my specialty. My research is basically on shallow reef corals in Southeast Asia and the world. So that's me. Thank you very much.
Thank you. Good afternoon, everyone. My name is Swee Hee. I'm the assistant head of the museum. I do whatever Darren, Tan Wei, Susan, and Peter tells me to do, okay? But usually, typically, I'm being bothered by the facilities, the operations, and the gallery of the museum. So these are my daily bugbears I do worry about. Thank you.
Hi, good afternoon. I'm Martin. I'm a research associate at the Likhongchen Natural History Museum. I am working with in the Biodiversity Histories unit. And the unit seeks to contextualize our natural history specimens and collections using non-specimen materials. So you might have seen some of our recent publications where we try to bring in storytelling, narrative, and a more historical look at natural history and biodiversity history in Singapore. Thank you.
Okay, so we're going to conduct this as like a Q&A from myself to the panelists to discuss the topics I'm bringing up. But we also have a time where we would air your questions that you have submitted through Slido, or if you are here in an audience, we could solicit your questions later too. But I'm going to start with the first question. So today's theme for the symposium is specimens, stories, and science. So as what some of our speakers have talked about, so maybe my first question is, how do you see the role of the museums in bridging these three items, specimens, science, and stories together for both researchers and the public? Maybe I'll start with Tan Wei.
Thank you, Marcus. I think these three topics are the first word on that is probably the foundation of all of that, right? It's the specimens that we are concerned with, and I think throughout the talks today, you have heard that the specimens are where we start our research. Ultimately, we are interested in understanding biodiversity in Singapore, regionally, and globally, and that forms the basis of most of what we try to know for various groups of animals in particular. But behind all of that lies, stories in which how the specimens came to us, and especially for specimens that were collected before Likhongchen Natural History Museum, even before RMBR, Raffles Museum of Biodiversity Research, and in the 19th century and so on. So all of that information is really important because that gives us an understanding of biodiversity and also the associated factors that result in either increase or decrease in diversity, richness, and so on in terms of biodiversity trends over time. So the stories are really important, but when we are talking about, of course, the science, the half of that museum that are doing the research, we are all engaged in the science, and so we hope to communicate that to the public, because ultimately the specimens don't belong to us. We are really just the caretakers of the specimens. We hope to communicate the science and those stories to the public, and we do that through the papers we publish. So that's most of them are out in the public space, but also through programs that are run by our outreach and education unit, our section, and also through the gallery that we invite members of the public to come in to learn about biodiversity. So that's how we connect these three topics. But ultimately, we think that the specimens are really important. So that's why we devote so much time and energy to maintaining the conditions of those specimens. So apart from all of that, all of those things you have heard from the researchers, is that actually a lot of time is also spent trying to maintain that collection, and that has been going on since the 19th century, through the time where our specimens were, possibly in peril in the 70s, but now hopefully we are in a better place now where we can move on from just taking care of them to learning about them and then to letting everyone know about what those are. Thank you.
Great. Dr. Tan, do you have anything to add on to that?
Since I manage the gallery, so to me, the job is made easier because we have so many specimens. Right, for me, I feel it was easier to tell a story through the physical media, right? As opposed to, like, for example, you want to mount an exhibition on culture of Singapore, then it's kind of abstract, so it's a bit difficult. So I think I'm quite fortunate in the sense that we are working in the museum with such a large collection of specimens that makes the translation of the information we want to bring out to the public much easier, right? And of course, we are very blessed to have very good curators who have been curating our specimens for so many years in good condition, and also a very active education and outreach group who helps the gallery side to push out the information and use it as an educational space. So I think that's how I look at the specimens from the gallery point of view.
I think it's a great time to be a natural history museum. It's a great time to be doing research, and especially heritage and history-related research in Singapore at this time. We've often talked about it with some of our other colleagues from other museums, that in 2019 there was sort of a natural history turn in how heritage was presented to the public. So you might remember the Raffles Revisited exhibition at the ACM. You might remember the Human Nature exhibition at NLB and our Bicentennial 200 exhibition. And I think Singapore has come to a point where we have had enough of looking at big men, big characters, big women. We've done that whole thing. We've moved on to other parts of our heritage, and maybe it's time to look at the non-human. And I think a lot of what the content that was presented during the bicentennial was to actually look at nature, the environment, and the non-human, so the plants and the animals that share this island with us. Not just now, but all the way back into the past. And I think because of that, and the confluence also of our previous head, Professor Peter Ng, and our current head, Prof. Yeo, through I think the SIGNIFY project that you have heard about today, to see that specimens also have their own stories. All right, I think we've all come to that realization that specimens have that story, they have a history, and we should, very much make use of that, and to bring those stories, draw those stories out of the specimens that we work with. So not just in our own collections, but also in other collections. And to also, just to add on, to look at non-specimen artifacts that can potentially enrich that story.
Yeah, thank you so much. This is a great summary between the three panelists talking about how specimens research can bring things together and how we are interconnected, not just, we don't exist alone, but with other museums too, and there's a greater wealth of resources and stories beyond the specimens. But since this is a research symposium, I want to tie it back to research that the museum does. So Tan Hui here, he's not only the deputy head of the museum, but he's also a professor at the Department of Biological Sciences at NUS. So I want to ask you, Tan Hui, in addition to doing research at the university and at the museum, but you also manage the research group at the museum here. So could you tell us what are the differences between the two for you, running a lab at the university and managing the museum's research group? What's the difference, and what are some of the challenges, or what is most rewarding?
Thanks. Yeah, this is an interesting question because I can say I run the research lab in the Department of Biological Sciences, but I really don't run the research in the museum because everybody runs on their own, right? And really the research collections and curriculum section are made out of a bunch of very talented and independent scientists. They are all, in a sense, principal investigators in their own right. They run their own research programs. I think one of the main difference is that in the museum, we're very much focused on specimen-based research and biodiversity, and so all of them work on their individual taxa. Anything that is related to the taxa, they work on, and they do it. They have the freedom to do that, in fact. And I think as a natural history museum, we are very lucky that we have the resources to support our researchers to work on those topics. Because as you have heard from many speakers, that biodiversity and specimen-based research is not something that is very easily fundable at the national level, especially when we are looking at more topics that are related to impacts, to environmental stress, and so on. So back in the department where I do a bit more of the running, our research is a bit broader in the sense that we go for grants that we might be funded for, right? So it may not necessarily be specimen-based, but really we have to pitch our research in a specific way. So we don't have that freedom, and I think so. Right now, the museum, we still have that freedom to pursue biodiversity research, and as our management and also NUS, we hope that to preserve that and to be able to maintain that focus in the next decades to come. In terms of, I think, the people that are working there in the labs, because we have students, postdocs, and staff who are in various stages of their education, in their research and growth. Actually, the greatest satisfaction is in seeing them move out, right? They graduate and then they go out and get great jobs and do their research somewhere else. But on the contrary, in the museum, we gain the satisfaction in having our staff stay and continue to contribute in NUS and in the museum. So I think the focus is very different in the research lab. Is we hope that they go out. We don't want to keep them. We want them to go out and do great things. In the museum, we hope that they do great things in the museum and contribute to biodiversity research in that way. Yeah. So those are the main differences, I think.
Cool. That's great. So if I ask you to put on your educator's hat now and you give a letter grade for the museum's research performance in the last 10 years, how would you grade it? What would you say was good, and what would you say needs improvement?
Okay, so I have to be quite politically correct here, right? But okay, I'll give it an A minus, and I'll give you a couple of reasons why. Okay, I would have given you A plus. But in Singapore, we never say we are A+. We are always A, so that there's room for improvement. And there's indeed room for improvement, right? I think we are still a growing institution. We are at 50 staff. So if you think about Natural History Museum in London, they have 300 staff just doing research, right? Their science-based staff is 300 strong. But we are not far behind in terms of our research progress. So I'll give it an A minus because of the potential we have, and the fact that if you are comparing ourselves to many of these global institutions, we still have some way to go. Just for example, we actually produce a lot of research, right? Every year we publish around 100 papers, and in a wide variety of topics, particularly in taxonomy. If you compare that to NHM, Natural History Museum UK, they publish around 700-800. So it's a lot more, but remember, we only have about 20 staff that are publishing those 100 papers. NHM, they have 300 staff publishing those 700-800 papers. So we are punching above our weight. But that's the point. The constraint on us is our size. And we hope to grow that size physically, as in terms of the museum space to accommodate more specimens, and also for more talents that we can bring in on board to study Southeast Asian biodiversity. And then we will get to A, and then hopefully get to A+ over the next few decades. So yeah, thanks.
fantastic. So we feel better about our research now. So my next question, still on what we've done for research in the last 10 years, is about how we are connecting or displaying this to the public. So Dr. Tan Swee Hee here leads a team that curated the 10th anniversary exhibition that's going on right now at the museum in the temporary gallery area. So I would like to ask Swee Hee, going through our research in the past 10 years, what was the one thing that maybe moved you the most or excited you the most, and how did you connect with it and aim to communicate this with the public in the exhibition?
When you say moved, I think traumatized. And the answer I'm going to give you will be the sperm whale that we got shortly after we opened in 2015. I still remember that day we were having a small management meeting in the conference room, and you burst into the conference room saying, "There's a whale off Jurong Island." Well, then I look at my watch, oh, it's Friday, there goes the weekend. And sure enough, that started a 70-day of craziness that I think none of us will ever forget. Everything was OJT, and there was so many moving parts, unknowns, variables that we do not know whether we can even overcome. But thank goodness for whatever reason, and because of good connections, we managed to push through it. And, we even got it up on display to be launched during SG50. So to me, it was the whole process of getting the whale off the sea onto land, butcher it, clean it up, bring it to the museum, build a mount for it, and stage an exhibition around it. That was something I will never forget for the rest of my life. No thanks to the PTSD left over from the smell. So yeah, that's something I always remember.
Thank you. Yeah, that was really a big project, and really everyone in the museum chipped in at that point of time, and it was great. And like I think what Prof. Peter Ng said just now, whether he would do it all over again, he said, on his slide. Yeah, so now I don't know if you want to do it all over again. But for Martin, so you have worn many hats in the museum, being the editorial team in the journal, being secretariat for ICZN, what Alex is doing now. But now you are working in biodiversity histories. So, but with most of the contemporary researchers striving to find new species knowledge, make new information, but instead you're looking into the past for research. So, why is looking into the past important?
I like to see it as kind of a mirror. So the really great research team that we work with closely is looking into the future. So it's a lot of, collecting, assessing what we have currently, through all the expeditions that we're doing, not just in Singapore, but outside of Singapore with partners. In thinking about the future, preparing for the future, inventorying the biodiversity, to think about the future. But at the same time, it's not just biodiversity histories. I think we acknowledge, that all our colleagues, we help each other, we work together. But the museum as a whole can also play a role as a mirror to look backwards into time. And I think this is very clear with the SIGNIFY project. Through the project, as they have gone to overseas museums to look for Singapore material, they have actually found things that we did not know were found in Singapore, right? And they've been sitting in drawers for decades or even centuries. We've, for example, along the way uncovered some of the oldest natural history, those plants from 1796, through all these projects and efforts. And it's giving us a better idea of what happened in the past, what kinds of changes were made by man in the past. And, in the same vein as we are trying to discover more about what is out there, we are also trying to figure out what we have done in the past and how potentially that can make us better stewards of the environment. So it goes hand in hand. I think that's the most important thing. Yeah, to not necessarily think that one, they should complement each other. Yeah.
That's great. But what would you say to, I'm going to throw a curveball here. So what do you say to a naysayer who say, or ask a question, is this considered research? If all this is already in the drawers, already published in somewhere in the past already, is this considered research?
Well, it's certainly research. I think it straddles the humanities and the sciences. So I think the team, the biodiversity histories team, some of us, like myself, I've just enrolled at the Department of History. So we are trying to get a more interdisciplinary education, trying to be able to network better with our other colleagues. Basically this whole thing about breaking down silos. So I think it's not just, the lip service saying that we should be multidisciplinary, interdisciplinary, but actually doing it. So actually working with colleagues from across the campus, different parts of campus, and it's research. So I would be more. It's certainly research. I'll be careful whether to say it's science, whether it's humanities, social sciences. I think this is why I say it's a great time, because I think we can choose and we can push towards more interdisciplinary, more collaborative research that doesn't necessarily, even with new labels, new names, new fields. Yeah. So it's research. There's a lot of digging that involves, I guess what you mean by the naysayers is it, for example, reproducible? Is it, statistically significant? So I guess this is where, we are still trying in a way to figure out our way as well. There's always been the question, what is history? What is the study of history all about? What's the point of it? And I think in some ways we are also trying to answer that within the context of natural history museums and natural history collections. But we have realized that specimens actually do have a lot of information. Like I said, there are stories attached to specimens, the way they were collected, the collectors who collected them, the networks between those collectors, and the infrastructure that allowed all that collecting to take place, 300 years ago. Yeah. And I think that is what we want to find out, and to see how that can further inform the way we study specimens today. Yeah.
That's great. So with what you guys know or have plans already, would you be able to share with the audience here what we've been looking in the past, right? Last 10 years. Could we look into the future a little bit, and what do you know about the future, and what are you most excited about in your respective areas in the museum? Maybe I start from Martin and come back down.
So I think the SIGNIFY project is quite far along in terms of the funding for the current cycle. And I think it would be really, what I'm looking forward to is being able to look at all that material and to actually answer those questions that you asked earlier. What can we do with this data? So we have the data. There's thousands of specimens that have been digitized. They are on the website. They're freely available for everyone. But at this juncture, for us to actually come together as a museum, so not just the BH side, the biodiversity histories, but also the RCC side, everybody just come together, have a literally just talk about what we can do with that. And I think there's a lot of exciting potential for that, and I'm looking forward to that. Yeah, and of course in collaboration with other museums that hold these specimens as well. Yeah.
And the future for me will be dinosaurs.
Tell us more.
As you know, we will have a dinosaur exhibition co organized with Science Center. I'll invite everybody to please buy a ticket and go and see it. They will be featuring the Patagotitan majorum, which is supposedly one of the largest dinosaurs on Earth, stretching over 40 meters, which is almost twice the length of prints that we have. But very fortunately, with the strong support of the museum's management, we managed to invite a sauropod expert to the museum this year, right? And after 2 weeks of pottering around the 3 dinosaurs, unimaginable ways of measuring such a large organism, right? So he has collated his information, run it through his matrix, and I think there have been some rather interesting findings that we are not able to announce at this moment in time. Akan datang, right? So in future, I think once expert consolidates his research, I think there'll be more interesting things to report on the 3 sauropods that we have at the museum. Yep. Thank you.
So for RCC research collections and curriculum, as you've heard from the talks, there's been a lot of these barcoding, using DNA barcoding, but of course the foundation of that is the specimens built over 150 years and more. The barcode data actually serves as a good reference. So from now, actually, we can now move on to understand how do we better understand diversity, study biodiversity using this reference. So now we can use things like techniques like environmental DNA. We can use that metabarcoding approaches to characterize biodiversity in its totality rather than looking at individuals. Because now we have already that data as individuals. We can look at what is the diversity of a community, and with the database, I think we are in better state to do that. But also, we can harness a lot of new technologies associated with AI to better identify our specimens. I think that will be the next frontier for biodiversity science. So I think those are the two main projections that I think the museum will look for. Yeah.
Superb. So good things to come. Just watch the museum space. So one of the museum's goals is public engagement in biodiversity research and outreach, and that is why we have this symposium today. We also have the museum galleries that are geared towards this goal, and so are our outreach and education programs. So we are always interested to communicate to the public. So we have social media and a team that's communicating to the public, and so everything of this is organized towards that aim. And at the end of the day, we are interested on what your thoughts are, means the audience, as well as the general public, about our research efforts in the past decade. So we're going to try to achieve this 3 ways. One, the symposium organizers have gathered your questions via Slido and when you registered for the symposium. So we're going to go through the questions. I think we might have time for 3 to 5 questions today. And so let's go to that. Let me refresh the Slido first. Okay. Okay, let me go through some of these questions. Okay, maybe I'll go through this question that's been upvoted 6 times. So anyone in the panel can answer this or chime in if you wish to. The question is, what is one thing the LKCNHM can learn from other established natural history museums, and what's one thing you're proud of in the museum's journey so far? Anyone can answer this question or go through everyone.
I can start. So as I mentioned, we're not counting beans, right? How many publications that we have and so on. But I think there's a lot to learn from the established natural history museums, the positive things, and also learn what we shouldn't do, right? So it's important to, when we visit these institutions, to look at, for instance, how they organize their research. I think the advantage of some of these big institutions is that they have the flexibility to organize their staff and allow them to specialize a little bit more. I think currently a lot of our staff are doing a lot of different things, and so ultimately, if we are able to grow, if we are able to have research staff that focus a lot on the research, harnessing the power of the collections, but also staff who do both research and maintaining the collections, that would be very helpful in driving a lot of the research ahead. Currently, we do have our staff trying to maintain the collections, and right now we are at 1.2 million specimens across wet and dry collections. So that is a big number for 25 staff, right? Imagine every single bottle has to be checked every year, for instance. That's a large number of specimens that our staff have to check. And so if we are able to expand, then we have more flexibility to arrange to focus our staff on more different things. But also in terms of more broader management and running of the museum, there are, as you might see in the news, that a lot of natural history museums are refocusing away from science actually and moving towards more focusing on the gallery, and because those are the things that might bring in more revenue and so on. I hope we don't have to go that way. I think we still have to maintain a very rigorous research focus in order for our outreach to move forward. So as much as we want to learn from some of these institutions, we also know that a lot of the museums face challenges, and we want to learn from those challenges, and we hope that we still have those resources that we've been given because of the way the museum was set up, because of the way Prof. Leo Tan and Prof. Peter Ng have set up the museum in a certain way that allows us to harness those resources very, very effectively in order to carry out research and so on. So I think we are in a good position to grow, and we learn from those mistakes, those limitations that other museums have so far had, and I think we can move forward. Yeah.
To follow up on what Tan Wei said, a lot of natural history museums around the world seems to be under stress, or some are even being closed. So I think the museum is quite fortunate that we have a strong financial foundation built up by the previous management, so that in a way we can be somewhat autonomous in the way we manage our specimens. So unlike some museums, they are closing and getting rid of their specimens, we face the other problem, which is the collection is expanding and we are running out of space. So that suggests that active work is being done and specimens are being collected and added to the collection. I see that as a happy problem to deal with.
I think from the experience of working with older natural history museums overseas, I think one of the takeaways was that because we are so much younger and our collections are so much younger, we do not have that same richness in terms of the collections, very often, especially for exhibitions. So, when the museum in London wants to do something on Wallace, they have drawers upon drawers of specimens from Wallace, or from the Wallace period. Whereas for us, I think what has happened is we have, in a way, learned from them. We have learned, we can learn from the experience, but we have also learned to be very creative in terms of bringing non-specimen materials into conversation with the material that we already have. And I think that has been something that I have really appreciated. We sometimes feel like, it's so cool to be in London or Dublin where you have 100 specimens that are centuries old. But then on the flip side, of course, of that is that, we don't have a lot of the problems that they face as well. So, the SIGNIFY team, when they were in London, they were basically freezing because, the heating wasn't working, the building is not insulated. So that's the flip side of it. But basically it's been an exercise in how to be really resourceful, I think. Yeah.
Great. So from that question, I'm going to pivot to, there seems to be a lot of interest from the audience about trying to get involved in research or helping up the museum's science. So the next question, I'm going to combine a few questions together. People are interested, what can people do with interest to help the museum's research? One. And one specific one is, how do they help support the non-fab science that Prof. Peter Ng mentioned, right? That do not see direct, immediate impact as assessed today. Was anybody in the panel interested to take up this question?
Okay, so all of us have our phones, and I think your phone is really, really powerful. There are two functions there that are really powerful, right? There's the camera, and then there's the social media, right? Those two things put together. If you are going out and you are taking photos of wildlife, I think that really broadly helps in broadcasting the amazing attributes of biodiversity, even in Singapore, right? And everywhere you go, you will see new things that you have not seen before, and you might want to learn about them. I think if you post them, there will be natural history nerds who will tell you what they are. So post it up, and someone will. I think that generates a lot of interest, and I think if we are able to keep that going, it will be really powerful. And sometimes those things can be new, and they get to us, and that's when you become involved in the science, right? So, and that's what the non-fab science we are talking about, right? Those that, if we are not able, we don't have resources to go out and collect because we don't have the funding, for instance, not high impact science, you can be part of it, and you can be the initiator of that process. So I think get out there and take photos and post them. I think that really actually helps because eventually they get to us or to scientists anywhere and around the world, and then you can be part of the science.
Yeah, I'd like to add on to that. So you don't even need to really get out there, just get your phone out. So recently, if you look at our Singapore biodiversity records, there was actually a beetle that was found by a friend of mine in his apartment in Marine Parade, right? And the beetle happened to fly into his kitchen during COVID. He took a photo and he showed it to me 3 years later, right? And I'm like, dude, where's the specimen? But it was new, and it was a new specimen. It was a first record for Singapore. It was in the same genus as one that Maosheng actually found in the Gardens by the Bay. Yeah. So this genus seems to be making its way into Singapore, right? And so you don't even need to leave your house, right? Just get your phone out. We have the Biodiversity of Singapore's website, that can be useful for you to actually compare what you have, do a first cut. And there are ways of learning, but really, like Tan Wei said, just to get it out, put that information on social media, bug our bug researchers. I know they're going to kill me later, but yeah, get in touch. I want to tell you about this little piece of paper that I just was shown by a researcher. So in the 50s or 60s, when Michael Tweedy was still director of the Raffles Museum, a teacher from a school in Pulau Tekong actually wrote a letter to Michael Tweedy on behalf of his student, and they basically sat down together and drew this butterfly that they found and sent this letter to Tweedy asking for, what do you think? Is it rare? Should we give you the specimen? And the letter today is at the ACM, right? So my friend was going through the archives and happened to come across it. So these things have, it's not new, but what we do, what is really new is the technology. Everyone has a phone in their pocket, right? Capture the data and share it, whether it's on forums, whether it's directly through researchers who you have gotten to know. I think that's the way forward. Yeah, and I would like to see a lot more non-specimen things as well for selfish reasons. Thank you.
Fantastic. So I'm mindful of the time that we have, so I'm going to quickly sum up the session today. So we learned about what the museum does, how it's managed in terms of research, how Dr. Tan that manages the gallery translates research into into gallery specimens, and how we looked into the past to learn more about it and generate new information, and how even people who are interested in natural history can actually contribute to creating more information just by using our phones and chatting with a curator, for example. So we are counting on you to support the museum's science in any way that you can, especially when it comes to social media, when it comes to our exhibition. So I'm just going to go through several things that are happening. First, there is the dinosaur exhibition that is going to be launched soon. There is the outreach and education units programs that you can find more information from on Facebook, as well as the museum's Instagram and website. And there is the existing 10th anniversary exhibition in the gallery that you can go visit to learn more about everything that's talked about today. So I would like to thank everyone for being present or coming to the museum's research symposium. Hope to see you again at the next one. Thanks very much, everyone. To the panelists here and the organizers for the symposium as well. Please give them a hand, please.
What an intriguing panel discussion that was. And with that, we've come to the end of the symposium. We would like to extend our heartfelt thanks to all our speakers and panelists for sharing your work and insights with us. And a big thank you to all of you for being such an engaging audience and joining us to celebrate the museum's 10th anniversary. Do note that the photo booth will still be around until about 5:30 p.m. More importantly, remember to use your complimentary museum admission ticket in your pouches. They will be valid till the end of this year. And with that, goodbye, take care, and keep exploring.